Okay, we are live and I will start the webinar so that attendees can start joining. Okay, we are live and I will start the webinar. Oh, sorry. So that people know there is a little delay between the Zoom meeting and the YouTube broadcasting. It's about 10-15 seconds, something like that.
Great.
Yes, say.
Could people turn off their cameras and their microphones?
Okay, and then we start. Right. Perfect.
Sol, nai dusenean.
Sorry, Rafa?
Nai dusenean.
Ah, nahi dezunean.
Dusenean. Also.
Bye.
Bueno, let's get started. Good afternoon, good evening, or good morning, wherever you are in the world. My name is Rafael Yuste. I'm the professor at Columbia University and director of the Neurotechnology Center. And together with my co-organizer, Aitzol García Echarri from the Donostia International Physics Center in San Sebastian in Spain, we welcome you to this third edition of the NanoNeuro Symposium. So what is NanoNeuro? So NanoNeuro is the intersection of nanoscience and neuroscience. And there's a lot of very potentially fruitful connection between these two fields that have evolved completely independently from each other, but they are experiencing a revolutionary growth. Among the interactions between nanoscience and neuroscience is the possibility of using novel nanomaterials as methods to develop new tools for neuroscience. Okay, so and why is that important? Why is NanoNeuro important? Well, as you know, understanding how the brain works is one of the frontiers of modern science. And the reason why we still don't understand how the brain works, or one of the reasons, some of us think has to do with the fact that we don't have good enough methods to record the activity from vast numbers of neurons in the brain and to modulate or change the activity from these large neural circuits. And the push to build new methods, so neurotechnology, started a decade ago with the launching of the US Brain Initiative, which was announced by President Obama, which is a large-scale project to develop methods for neuroscience, again, neurotechnology to either record brain activity, to image brain activity, to image structure of the brain, and to change all of that, to manipulate the brain. And this new technology could be important not just for neuroscience, but also for the clinic, for medicine, and also it could lead to many new economic and commercial developments. So nanoscience has a lot to offer, because for the last 20 years, nanoscientists have explored and created new types of material that have fascinating new physical properties. And in many cases, these new materials, either nanoparticles or probes, are ideal platforms for the development of methods to measure or manipulate biological tissue or brain tissue. So we think this is a revolution waiting to happen. It has not happened yet. There's been some interesting work. And this leads us to this meeting. So this is the third edition of a meeting. We started it, again, with the intention to build bridges between nanoscientists and neuroscientists, and in the hopes of creating perhaps in the future a new field that you could call NanoNeuro. And to alert also the science in general as large, and also funding agencies, that there's a lot of potential opportunities here. So because of COVID, the meeting was done online, and we had very positive feedback. So this is why we're doing it again this year. So the meeting is possible thanks to the sponsorship of the three institutions: the New Technology Center at Columbia University, the DIPC in San Sebastian, Spain, and also the Chen Foundation, whose general support has made this meeting possible. Also at Columbia, James Holland and Lax Steinberg have played a critical role in the organization of this meeting. So let me quickly tell you what the meeting is going to entail. We'll have two sessions. The first one focused mostly on nano, not exclusively, and Aitzol will be the chair. And the second one focused mostly on neuro, and I will be the chair. There will be a break between both sessions, but it's just a half-an-hour break just to go and get a cup of coffee and to go to the bathroom. And then we have a series of speakers. The speakers will have 25 minutes to make the presentation, and this will be followed by a 5-minute Q&A. And also at the end of each session, the speakers will join Aitzol and me for a half-an-hour panel discussion on the topics that emerged during the talks. At the end of the meeting, we're going to have a keynote lecture by Alice Ting, who joined us from Stanford University. And again, this will happen after the second session, and it will be followed by another panel discussion with the speakers of the second session. For those of you in the audience, we welcome your questions. There are different ways you can send us questions. If you're following the meeting through Zoom, you can type in your question in the Q&A. If you're following the meeting through YouTube, you can also type in your question in the comment section of the YouTube platform, and those questions that you type there will be relayed to us in the Zoom platform. Speakers are encouraged to ask questions to other speakers, and they can do that either by raising their hand and unmuting themselves and just asking the question directly, or they can also use the chat function that the panelists have access to type in the question. And I think with this, we've got all the instructions out of the way and we're ready to jump in. So I pass the mic to Aitzol. Aitzol, you can take it from here.
Okay, so welcome everyone from my side as well. Ongi etorriak, NanoNeuro 2022 konferentziara. For those of you who are international, which are most of you, that was Basque, a language that we speak in the Basque Country, a tiny region in Spain. And I just wanted to say a couple words on how we started this adventure of NanoNeuro, Rafa and me, not only about the conference, but our collaboration in general, as an example of, I don't know, a story of initial success, let's put it that way. So Rafa and me met in 2018, and we had, I don't know, common interests and common curiosities. And so we started looking for projects that we could develop together, and we came up with this application of nanoscience to the neurosciences. Why? Because I am a nanoscientist. I do nanophotonics, the interaction of light with nanoparticles, and Rafa is a neuroscientist. So that was 2018. Today we are in 2022. And apart from having organized 3 wonderful conferences, with 2 in the past and 1 happening right now, in these 4 years, we actually coordinated a group of 7 research institutes, and we are developing a variety of projects in this field that we call NanoNeuro. And these projects are giving rise to quite a few PhD theses, etc., etc. So we are very happy research-wise as scientists about these projects that we are developing these days. And today we are extremely happy to welcome you all to this NanoNeuro conference. By the way, one last thing, I wanted to send a big thanks to Teresa Zelaya, Susana Lauera, Javier Alfaro, and Escobar and Marek, because they collaborated preparing this meeting, contacting every single invited speaker, proposing names, etc., etc. And this meeting wouldn't be possible without you guys. So thank you very much. And with these welcome words, and since we are 5 minutes late already, and we still didn't even start the meeting, let me introduce to you to Guillaume Bafou. Guillaume Bafou is a CNRS researcher at Institut Fresnel in Marseille. And Guillaume, I know him from the field of nanophotonics, which is basically my own research background. Guillaume and I don't know each other personally, but we have 1 trillion friends in common and a lot of common interests. Guillaume wrote the book on thermoplasmonics, I would say, a book that we use in our lab on a daily basis. And today he will be talking about phase imaging for thermoplasmonics, how to measure temperatures in plasmonic nanoparticles, and biomicroscopy. And since we are running very late, Guillaume, I will give the mic to you, and we see each other in 25 minutes again for questions. See.
Okay, thank you for the introduction and the invitation. Yeah, so I'm glad to participate in this NanoNeuro symposium. My background is indeed nanophotonics, but I have been working since recently in biology as well and in neurobiology. So, and in particular, today the quantitative phase microscopy technique I'll be focusing on has been applied. We applied it to nano and neuro. So this is what I would like to show you. And more precisely, so I will first describe this quantitative phase microscopy technique that is very special. And then I will explain, oh, sorry, this is not presentation mode. And then I will explain how this technique, we applied it to image cells with a very good contrast. Then how we used it as a temperature microscopy technique, and in particular, how we can map the temperature within living cells. Also, we applied it in nanophotonics to characterize nanoparticles. And finally, more recently, we image neurons in culture using this technique to measure dry mass, to perform some mass measurements of neurons and neurites. So this is basically the outline of my talk. So what is quantitative phase microscopy? So this is your sample on your microscope. This is, for instance, so this is the side view of your sample. This is, for instance, a living cell deposited on glass. If you send light on top of this cell, the light crosses your cell. You have a wavefront distortion. So the wavefront of the light is distorted because the light travels slower within the cell. So this is a distorted wavefront, which is delta L, and this is the expression, and D is the local thickness of your cell. And phi is a phase, and phase and wavefront, they are related using this equation here. So there are many different quantitative phase microscopies. Some of them, most of them, they measure phi, they measure the phase using interferometry. The technique we are using, it's not measuring the phase profile, it's rather measuring the wavefront distortion itself. So it's more a wavefront microscopy technique. But it can do exactly the same things as quantitative phase microscopies. So this is a setup. So it's like a normal microscope you have in any biolab. You have a light source from the top, your sample, objective lens, tube lens, and your camera. So you may say, yes, this is exactly like a normal microscope. Yes, the only difference between a normal microscope and our microscope is the presence of a two-dimensional grating, a 2D grating or a cross grating, which looks like that. And you place it just in front of the camera, like 1 millimeter from the sensor of the camera. And this configuration enables you to measure the phase of light or the wavefront of a light beam, provided the grating is spatial and with black lines, that you can see here, defining holes, transparent holes, and with a checkerboard pattern with 0 and pi phase shifts. If you do that, then you can map the wavefronts. So what is the working principle? Well, if you place this grating in front of your camera, you have this kind of image. So you can recognize maybe the nucleus of the cell. So the image doesn't look pretty because this is a raw image. It's not processed. But if you zoom in, you can see dots, bright dots. And each dot corresponds to one transparent square of the grating. Then if your wavefront gets distorted, all these dots, they will move. And this is where the information on the wavefront distortion is. Another way to explain it with a side view of what's happening between the grating here and the camera here. So each hole creates like a beam, like a light beam. And here, this is where you have the bright spots. So I have only drawn like 8 holes, but the real grating is much bigger than that. This is a wavefront on the left, crossing the grating and arriving on the camera. If this wavefront is tilted, distorted, these bright spots on the camera will move, shrink, expand. And this is how you can retrieve the local orientation of the wavefront, the gradient of the wavefront. So to summarize, this is a raw image. From this raw image, so here this is like a micro bead. From this raw image, the intensity of the spots gives the image intensity, and the position of the spots gives the wavefront. This is where the information is in the raw image. And then you can place anything you want on your microscope. Here you have a microbead, but you can put like plasmonic nanoparticles, neurons, bacteria, anything you want to study them in phase and intensity. Something we never show in presentation or even in articles is the noise of the images. We usually don't do that. I do it here because the noise level of this cross-grating phase microscopy is particularly low. So here, this is a standard deviation of the noise amplitude you have on a CGM image. And this value here, 0.039 nanometers, it's a fraction of hydrogen atom. So what it means is that if your wavefront distortion, if your wavefront is distorted by an amount that is smaller than, that is on the order of a hydrogen atom, we see it, we can detect it. So this is typically the kind of image you can have when you oh, yesterday the movie was working fine, doesn't moving. I'm not sure you see it moving, I don't see it moving. Anyway, so on the left and on the right, you have exactly the same image, the same cell. We just here high-pass filter will just apply on the right image. And you can see many details, including the mitochondria. Here, the long wires you see are mitochondria moving along the microtubules. Another comment. So maybe you know this technique. This technique is not well known. It has been invented like 20 years ago, so it's very old, but it's not well known, although it's very powerful. If you know it, maybe you know it under another name, which is quadri-wave lateral sharing interferometry, or QWLSI. So I prefer to use this name for the sake of simplicity, and also because in biology, usually my experience is that this name is quite reluctant. Especially interferometry gives the feeling that you have to make two beams interfere to get an image. It's not the case. From what I have explained, if you understood everything I said, you just have to plug a camera in a microscope, and that's it. And you get the images I have shown. So it's very simple. And this name, the complexity of this name is not an image of the simplicity of the technique. Okay, so if you want to know more about this technique, I'll focus now on the applications. But if you want to know more, I encourage you to read these two papers we recently published. The first one on the working principle and on the algorithm to process the interferogram into phase and intensity images. And also the second article where we focus on the accuracy and the noise we can expect using a CGM. In any case, all the algorithm related to these two papers, they are on my GitHub account. So let's focus now on how this technique, this very simple camera, can be used. So first, we used it as a temperature microscopy technique. What I have been doing during maybe 10 years and more, I have been heating gold nanoparticles using light. If you shine gold nanoparticles, they absorb light and they become nano sources of heat. So you can heat easily at the nanoscale, at the microscale. In this kind of application, the challenge is not really to heat. This is easy. The challenge is more to measure the temperature. This is complicated because this is happening at the microscale. So one way to do it, and this is the technique we developed like 10 years ago. One way to do it is to map the wavefront, light wavefront distorted by the temperature field. If you send light through this system, you have a liquid around. When you hit your particle with a laser, you have a variation of the refractive index in the liquid, which creates like a thermal lens, which distorts this wavefront. And we measure this wavefront distortion using CGM. This is still not the temperature profile, okay? This is a wavefront distortion created by a three-dimensional temperature profile. But we can retrieve the temperature profile using an inversion algorithm. This is what is shown here. This is a single nanoparticle. We hit it with a laser. This is a raw data wavefront distortion from which we can retrieve the temperature map, but also the heat power density. And this is working for one nanoparticle, but also for any system that is heating even many nanoparticles. So with this technique, we have done many things in physics, chemistry, and biology. I just want to focus on one of these applications related to biology. We have been culturing cells on top of this layer of cold nanoparticles to heat them, to heat cells, single cells in culture. So we hit with a laser, and on top of this system, we have another light to illuminate the scene. And these are the three kinds of images we can acquire in parallel using this technique. So the first image you see here, this is a wavefront distortion. This gives a very high contrast image of your cell and quantitatively. The second image, this is a temperature profile. So as you can see, it's hot. It's hot because we are heating with a laser. There are nanoparticles everywhere. You don't see them because they are very small, but you have a laser beam this size, this big. And this is a resulting temperature profile, which is measured using exactly the same camera as for this image, also using wavefront distortion measurements. And just because this technique is label-free, you don't have to label your cell with fluorescent molecules. You have free hands to do in parallel fluorescence microscopy. And this is what we have done in parallel. So what did we do? We wanted to investigate how the cell responds, fights against the stress created by a temperature increase. When biologists, they want to do that, and this is what we have done, they label transcription factors within the nucleus that are responsible for the production of heat shock proteins. And when the cell is stressed, these transcription factors, they agglomerate and they create granules within the cells. So yeah, yesterday the movie was working, but it's not working at all now. It's a shame. Okay, so I tell you what you are supposed to see. You are supposed to see the appearance of granules within the cell. So very bright fluorescent granules within the cell here. And this is when the laser is on, and when the laser is off, they disappear. So what we have done, so this is what biologists do, but they do it by hitting the stage. Usually they don't hit one cell at a time with a laser, they hit the stage. And this creates a huge inertia. Usually you have to wait like 10 minutes before reaching the target temperature. So what we have done is to follow the intensity of the spots as a function of time. And we switched on the heating laser for 1 minute and follow these two granules intensity, 1 and 2, as a function of time for different laser powers, corresponding to temperature increases of 39, 42, and 44. And interestingly, after a few seconds, you already have the granules appearing within the nucleus, which means that the cell can react very fast to a stress. This time scale of a few seconds, you cannot investigate it if you hit your microscope stage. To investigate this time scale, you have to hit at the microscale. It's the only way to have a short time scale. So this is what we have shown here, the benefit we have evidenced. And interestingly, the more you heat and the more time it takes for the cell to recover its, to fix the stress, as you can see here. So this was a work of my previous PhD students. Okay, so another application, this time specifically related to nanophotonics. We have been imaging nanoparticles. So no longer temperature profiles or living cells. We have been imaging nanoparticles. So you remember what I have said, you can image wavefront distortion due to a temperature gradient, but what you can do is also image wavefront distortion due to the nanoparticle itself. And if you do that, you have like an airy pattern, like with diffraction rings. So the images are beautiful. I like them a lot, but it's not the only interest. There is also a scientific interest, and which is this one. If you consider these two images, intensity and phase, and you multiply them pixel by pixel, more precisely, if you multiply the square root of the first one by the complex exponential of the second one, and if you sum all the pixels of the resulting image, you get a number, a complex number, which is nothing but the optical complex polarizability of the particle. So this is the most fundamental feature you may want to know on a particle when you want to do some metrology, because once you know this polarizability, alpha, you can retrieve all the absorption cross-sections. For instance, here, these are the measurements, the circles are the measurements of the absorption cross-section of a 100 nanometer gold nanosphere as a function of the wavelengths compared with theory. And you can see that we have a very good match between theory and experiment. So this is like a very convenient tool, metrology tool in nanophotonics, because usually when you want to measure these three cross-sections, you have to use three different setups. Here with one single image, you have all these features at the same time. And the last application I would like to show you concern neurons and neural cells in general. So this technique can be used, and in general, any quantitative phase microscopy technique can be used to measure masses, and especially masses of cells. So I don't know if you know this application. This is maybe the strongest benefit of quantitative phase microscopies. So this is just to explain the image of a bacteria. Delta is the wavefront distortion. If you sum all the pixels of a wavefront distortion image, you get what we call the optical volume. And it turns out that this optical volume is proportional to the dry mass of the cell, up to a constant factor, which is 0.18. So if you sum all the pixels of the image, you basically have the mass of your cell. This is a very interesting feature that enables you to follow the dynamics, the evolution, the growth of cells in biology. So we use CGM to do that. And because of the very high sensitivity, we could follow the mass of very tiny tenuous objects in biology. And our attention, we focused it on neurites because they are very small. So this is a basic growth of a neuron from the early hours to 7 hours here. So at the beginning, we have like a lamellipodium. After a few hours, you have neurites. And we focus on the very first hours of development of a neuron. Oh, this movie is working fine. So this is a typical CGM image you have over time. So you see the neural cells everywhere, and you see what is usually not clearly visible on normal images. You see like the lamellipodium of the cells and even the neurites growing, moving. You can see some waves within these cells. So what I just said is that if you sum all the pixels of any part of this field of view, you get a mass. So we have done that on specific neurites. So you have the soma that is here. So what we did is to segment the soma to isolate only the neurites. Oh, this one is working as well, right? And so this is the typical time lapse over 15 hours of 2 neural cells interacting. So I will speed up a bit. I will go here. So this second cell you see here, you have a neurite that was born here. It is growing and growing, and at the end it will reach the other one. And what we did is to segment the neurites to map the dry mass, the touch at the end. So we focus on this one. You see the upper one that touched the second one. I'll show you in the next slide the dry mass of this neurite here. So this is the dry mass as a function of time during 15 hours. In green and in black, you have the length of the neurites in parallel. And what is very interesting with this precise neurite is that we have seen oscillations. So we talked to biologists, neurobiologists, and they were not aware of this oscillation within neurites. So it seems to be quite new and unknown. And what we want to highlight here is first the requirement to have efficient segmentation tools, because it's quite hard to segment these very tiny objects. And then to have a very highly sensitive technique, because otherwise you are overwhelmed with the noise of the image. But when you take care of these two things, then you have a sub-picogram sensitivity, and you can be sensitive to some oscillations of neurites like that. So now we are at the stage where we want to understand this kind of oscillation, where they come from. Why do we see that only on very specific neurons and not on anyone? So this was the work of my previous PhD student. Okay, so I'm done with all the applications I wanted to describe today. So just to sum up, with this cross-grating phase microscopy, you can image cells with a very high contrast and very good sensitivity. You can use it as a temperature microscopy technique, and in particular in biology. You can characterize nanoparticles and measure dry masses in biology. So at the Fresnel Institute in Marseille, in France, where I come from, here are all the people involved in all the work I presented today and my collaborations. And thank you all for your attention.
Thanks a lot, Guillaume. That was an excellent talk. And as the chair, I need to say thanks a lot as well for keeping up with the time. Your talk were exactly 25 minutes. So we already have a couple questions online. And I don't know if you can read them, but I will read them for you no matter what, for the audience. Okay, there is a very interesting question here that I also had. An anonymous attendee is asking, why is the optical distortion proportional to the dry weight? Thanks. So what's the fundamental behind it? So
this is because the refractive index is directly related to the density, to the mass density. The denser the material and the higher the refractive index. And the proportionality is true for a given range of material. For instance, in biology, this alpha factor I mentioned is 0.18. But for other kinds of materials, it's not. So as far as you stick to biology, it's fine. It's 0.18.
All right, so we have a couple more questions. So I see one by Marek, a co-worker of us here in San Sebastian and Donostia. He says, in relation to the first application, thermometry, to what extent the chemically complex environment in living cells can affect the determination of local temperature close to the nanoparticle surface?
Yes, so I did not really explain the details, but indeed you have waveform distortion due to the temperature gradient, but also due to the cell. This is what I have shown. So you have the two contributions. But we have a trick. We actually measure the waveform distortion without heating and the waveform distortion with heating. And we subtract these two waveform distortion so that the contribution of the cell, we get rid of the contribution of the cell. This is how we get rid of that. And also we have to assume that the dN over dt, the variation of the refractive index of the cell as a function of temperature, is the same as the one of the medium, which is a good approximation. The refractive index is not the same, but the slope, the dN over dt, can be assumed to be quite the same.
I do have a question of my own related to that. So what happens with the dependence on the optical axis, let's say on C, let's say on the propagation axis? So are you integrating it and then get an average of the temperature in that direction, or?
When you image a cell, you get basically integration of the refractive index over the height. It's like a projection, okay?
Excellent.
When you image temperature, you also have a projection, but our algorithm can retrieve the temperature in 3 dimensions from a 2D map, provided the heat source is 2-dimensional, provided the gold nanoparticles, they are on the substrate. If they are floating in 3D, it doesn't work. But if they are 2D, we can retrieve temperature in 3D.
Excellent. And then probably the last questions that I will ask come from Alexandra Gavrilova. Sorry if I'm not pronouncing your last name right. So she's saying, thank you for your talk. I have a couple of questions. What are the limitations of usage of the heated gold nanoparticles? It's a little bit ambiguous, this question.
The limitations.
Yeah.
The limitation is actually in terms of laser power, I guess, it's making a bubble. This is the first limitation. At some point, you make a bubble in your liquid. But the gold nanoparticles, they are very stable, inert. They stick to glass. Yeah. They are biocompatible, so they don't affect your cells. So themselves, they don't give rise to any limitation.
Excellent. Then Alexandra as well is asking, is it possible to implement this technique not only in vitro, but also in vivo?
So I'll be honest with you, it's very complicated. No, I don't think so. Because you have, light has to cross the sample and has to feature a gentle distortion. If you have a scattering media, you just measure a mess. So imaging through the brain or within the brain, for instance, it's really only suited for application, I would say, on a cover slip in a microscope with thin tissue or cells.
Excellent. And I think we will leave the third question by Alexandra for the panel discussion because it touches on different aspects. So I'll keep it as a secret or as a surprise for the panel discussion. And if we're all fine, maybe we can move to our next talk. And so we can extend the time that we have for discussion in the panel discussion. Otherwise, we will have to cut it off quite strongly. So thank you so much, Guillaume. It was a wonderful talk and a wonderful discussion. See you later. Excellent. So our next speaker is Viola Vogel. She's a professor at the Department of Health Science and Technology at ETH Zurich. And if I'm not mistaken, in their lab, they work in applied mechanobiology. I've been checking a little bit Viola's history online, and I saw that she trained as a physicist. Then she did a postdoc in biophysics, And according to what she's working on these days, I would say that now she's mostly a biologist. And so I would say that her career is the perfect example of interdisciplinarity, or how you can take concepts from one field, apply it to another one, and then apply what you learned in this new field to a third field itself. So today she will talk to us about molecular mechanobiology of the extracellular matrix, healthy versus inflammatory cell niches. And with this, Viola, the mic is yours. So
thank you.
You want.
Thank you so much. Can you please activate my camera, because I can't do it from my side?
Oh, I don't know if I can do it from here, but I'll...
It was turned off from your end.
Rafa, maybe...
Why don't you try it now, Viola?
That's it. Okay,
You got it.
perfect, excellent.
Oh,
Wonderful.
thank you
Sorry
so
about
much.
that. Yeah.
Thank you so much for this wonderful invitation to speak here. Indeed, we developed a lot of nanotech tools in the last 20 years, from the molecular level to the cellular level and to the tissue level. And many of those can be applied to a huge variety of different cell types and tissue types. So as I go through my talk today, I would like to also introduce these tools and maybe stimulate ideas how you can use them differently to what we have shown so far. So certainly one of the key issues that bioengineers have dealt with since 20 years is to learn how to repair peripheral nerves. And here in this review, you see typical approaches that bioengineers have taken to finally guide neurons and neuron cells across the defects in order to connect the two sides and hopefully allow a healthy neuronal function again. So based on being in the same department as my colleague Martin Schwab, we started a collaboration a few years ago, and I would like to briefly report some of the findings that required nanotech tools. So Martin Schwab discovered NOGO-A in 1988. NOGO-A is an important axonal growth inhibitor in the adult and developing central nervous system. And NOGO-A is a transmembrane protein, and it has formed some loops, including this loop delta 20 that you see here also in the structure. And recent studies now show that if NOGO-A is presented, particularly the delta 20 subunit, it leads to inhibition and induces the depolymerization of actin. So it clearly has an impact on cell traction forces. And so in the moment, also Martin Schwab and others are developing now strategies to target NOGO-A to prevent scarring after neural damage. But every single nerve is accompanied by a blood vessel. And so it was very interesting to then also realize that NOGO-A is also a negative regulator of central nervous system angiogenesis. And what you always have when an angiogenic sprout is formed, you have a tip cell, and this tip cell needs to migrate or allow the sprout to pull itself into the surrounding matrix in order for it to grow. So we decided nanopillar platform, and different from most of the nanopillar platforms that were either made by like using electron beams or from PDMS, we use much stiffer SU8. And here you see a cell sitting on such a nanopillar array, and you see that cell traction forces can lead to the bending of these pillars. And since we know the Young's moduli of the pillars and their length, we can calculate traction forces. And so we asked whether endothelial cells, their adhesion and spreading on nanopillar arrays is impacted if we present the delta 20 fragment of NOGO-A. And indeed, you see the cells in red on fibronectin-coated surfaces. And then we also absorb delta 20. And in a dose-dependent manner, you see that delta 20 gradually reduces the contractility of these endothelial cells. We then asked whether the presence of fibers promotes or guides angiogenesis. And in order to ask even this question, we didn't want to use the typical spheres where endothelial cells grow into a matrix made of matrix gel or collagen gels, because these gels are so much denser than extracellular matrix fibers that you would find in healthy tissue. So instead, what we did, we pulled fibronectin fibers on substrates, in our case on stretchable substrates, and then took beads, again seeded with endothelial cells, and we placed these beads on these pulled fibronectin fibers. And here you can see some of these fibronectin fibers in red, the beads, and then endothelial cells that start to build sprouts. And to show you this more nicely in phase contrast, here you see the bead, and you start seeing how the first endothelial cells now leave the bead and pull themselves along the fibronectin fibers. Much more detailed analysis can then certainly be performed. So you see with lymphatic endothelial cells, as well as with UVECs, that sometimes single cells sprout off, and sometimes we see even collective growth, as shown here. And you see that the first cell pulls along the second cell, pulls along the third cell, until you have formed the first sprout on the fibers. Interesting is that the entire surface was coated with fibronectin. So the endothelial cells could have pulled also on the flat coating, but fibers were much more effective to guide the growth and to promote the growth of sprouts. We have learned in the last 20 years a lot about mechanobiology, how the stretching of proteins changes the function. We learned a lot about mechanoregulated cell signaling pathways at the cellular level. But then at the macroscopic level, we have so many observations that somehow mechanics really matter, but we do not really know how to probe it and quantify it, because we do not have nanoscale force sensors at the tissue level, nor do we have molecular strain sensors at the tissue level. And so we were thinking how we can best address this deficiency. So FRET probes don't work well because there's so much background fluorescence at the tissue level. We were asking whether we could use peptides to measure the strain of proteins. Essentially, the idea being that you destroy a multivalent binding motif by stretching your target. So Tom Barker tried to develop random peptide libraries using the same stretch assay that I've shown you before, in order to fish out antibodies that might sense and differentiate between stretched and relaxed fibronectin fibers. But to select peptides out of this library. But he didn't know where the epitopes were, how the mechanism of binding got regulated by tensional fibers, nor were the peptides stable in blood. So we have taken a totally different approach. We asked whether we can use bacterial adhesives, because bacteria and humans, the first bacteria showed up a billion billion years ago, fish already had very early fibronectin similar matrix. And so there's a long evolution of bacteria to invade hosts and to develop peptides to enable penetration into a wound site. So we discovered 10 years ago that the bacterial adhesin that S. aureus utilizes, which binds to fibronectin, because fibronectin is always present in wound sites, but in wound sites you cleave the fibers. And so we found, using steered molecular dynamics simulations, that it forms a very nice complex between relaxed fibronectin. But as soon as you start stretching fibronectin, the epitope is destroyed. So you really see, as soon as you just stretch the distances, you destroy the multivalent binding motif. And at the end, a low affinity binding occurs, but the peptide does not bind any longer with high affinity. So we were asking whether we can utilize this probe and develop it in a probe in order to measure fiber tension in healthy and diseased tissue niches. So what did we do? First, we verified, certainly using our stretch assay, that the peptide binds better to relaxed than to stretched fibers. Then we had developed already 20 years ago the very first thread probe ever used in order to show that cell-generated forces are sufficient to stretch proteins. And with this thread probe, for example, you see here the fibronectin matrix made by a fibroblast on a planar glass slide. Areas that are red show the areas where you have little fiber tension, and then the bluish green areas are highly tense fibronectin fibers. If you then label the peptide with a third color, you see that the peptide binds to areas where fibronectin fibers are much more relaxed. And then we could do a pixel by pixel comparison. And indeed, we use two totally different physical mechanisms, but the readout correlates very, very nicely. So different from single molecules, if you stretch them, as soon as you stretch them across an energy barrier into a new structural state, the strain of the protein determines whether the function has been switched or not. But fibronectin doesn't form periodically arranged fibers, and so we see a gradual change in peptide binding as function of the strain of these fibers. So what could we verify so far? For certain polyclonal antibody, we see that the binding epitopes get destroyed if you stretch fibers. We saw that fiber stretching destroys the collagen binding site of fibronectin, which is necessary to initiate the buildup of a collagen matrix in cell culture. And we saw that cytokine binding to stretched fibronectin fibers is different than to relaxed fibronectin fibers. And then also at the cellular level, because we now can grow single cells also on these fibers, we have seen that fibronectin fibers in early extracellular matrix are more stretched, and that osteogenesis is enhanced on stretched compared to relaxed fibers, and that this is EGFR regulated. So you see a clear motivation why we want to know the fibronectin fiber strain at the tissue level. And there was so much in the cancer biology community discussion about what physical characteristics are changing as a tumor evolves. And typically people have recorded and reported that tumor tissue gets much more rigid. And so we were very curious to see, are the fibronectin fibers in tumor tissue sections more stretched than healthy or more relaxed than healthy tissue in healthy tissues. And what we found to our surprise was tumors, as known, are very heterogeneous, but we see large areas where fibronectin fibers are structurally relaxed. And then we started correlating what cells are in the proximity. And surprisingly, you find a lot of myofibroblasts in the proximity of relaxed, not of stretched fibers. And that was again very surprising because myofibroblasts have an upregulated contractility, but they also have an upregulated synthesis of all kinds of ECM proteins. So they deposit a lot of collagen fibers. So what we find in the proximity of relaxed fibronectin fibers are thick cables of collagen, which we can visualize using second harmonic generation. Then we sliced up a lot of healthy organs, and in all the healthy organs that we sliced up, we essentially find that the fibronectin fibers are highly stretched. You do not see much peptide binding in contrast to what you've seen in the tumor tissues. Then we asked, what about lymph nodes? So lymph nodes, certainly they swell enormously upon an infection. So we were wondering whether we can measure a change in fibronectin fiber tension from naive lymph nodes and to adjuvant swollen lymph nodes. And if we do the stains, we saw very little change. So the fibronectin fibers in naive lymph nodes are under high tension. And then we looked at virally infected lymph nodes, and we found that virally infected lymph nodes show a lot of relaxed fibronectin. So is that relevant? And how could it potentially be relevant? In lymph nodes, you have cells, fibroblasts, they are called fibroblastic reticular cells. Different to typical fibroblasts, they also secrete a cytokine L7, which is absolutely necessary to maintain a healthy lymph node. And upon swelling, it was reported that the extracellular matrix is torn up by viral infections. Why is IL-7 so important? IL-7 promotes B cell and T cell proliferation and maturation. The lack of IL-7 causes immature immune cell arrests. IL-7 concentrations are very small in vivo, and IL-7 is more potent in a matrix-bound state. But despite the small amounts, B cells, T cells, dendritic cells, and lymphatic endothelial cells all compete for it. What we now found was, using our stretch assays, that IL-7 binding to fibronectin can be enhanced by fiber stretching. So that poses immediately the next question. Could it be that in naive lymph nodes, more IL-7 is bound and therefore presented to the immune cells compared to virally infected lymph nodes, and there are some indications that this might be true. So finally, we were asking, can we utilize this peptide not only on cryosections of tissues, but for theranostic applications? So we utilize this bacterial peptide to target relaxed fibronectin fibers and asked whether we can utilize it in living animals. In order to do this experiment, we radiolabeled the peptide, injected it in a mouse vein after testing that was stable in blood, and then indeed it accumulated in these tumors and was homing in diseased extracellular matrix. So with this, I would like to say that, and would like to argue that extracellular matrix fibers are much more than viscoelastic scaffolds to anchor cells. And we like to propose that cells not only feel the viscoelasticity of their environment, but that protein stretching plays a central role in regulating many additional functions. And in order to get beyond correlations, we are building up microtissue platforms in the moment in order to study many transformations, for example, of fibroblasts to myofibroblasts, where we can then, by purpose, change the medium to which these growing tissues are exposed. And we looked deeply into the myofibroblasts to fibroblast transition. Here you see in a microtissue that the growth front, which is only 20 micrometers thick, is very rich in alpha smooth muscle cells. But then the core includes fibroblasts. So we see a very sharp transition, and the fibroblasts are then found in the collagen-rich environment. And for these microtissues, the collagen-rich environment again shows binding of the peptide. So with this, I only briefly click through 3 more papers that are important, I think, to briefly highlight that if you have an injury, the first cells that create fibronectin are actually not the invading cells, but platelets themselves. We then saw that macrophage function is regulated if you don't allow them to spread, but to spatially confine them. And we recently could also show that nano-confinement allows to boost T cell activation if T cells are grown on nanoporous surfaces. With this, I would very much like to thank you for attending the lecture, and I'm looking forward to your questions. Thank you.
Thank you so much, Viola. So, oh, sorry, I'm getting a call. All right. So I don't see too many questions in the Q&A, but Rafa does have a question, so please go ahead.
Hi, Viola. What a beautiful piece of work, I have to tell you. So could someone make a GFP-based force sensor and put it in cells so that you can actually take a movie of the actual forces in the tissue? Is that a crazy idea, or is there hope?
So this was actually, those were ideas about 10 years ago, and people trying to make FRET mice. And reading out FRET, there's so much background fluorescence. And the background fluorescence is different from tissue section to tissue section. So calibrating a FRET ratio is so difficult to do. And that's why FRET works beautifully in cell culture, but not any longer at the tissue level.
Mm-hmm. And could you use the unfolding of GFP itself as some sort of force sensor? Is that possible? When you pull out the fluorophore, then obviously the water will quench it.
Many years ago, we simulated using steered molecular dynamics how the beta barrel actually unfolds. And indeed, you can easily pull out strands. And then we synthesized GFP that is fragmented, where certain strands are missing, and indeed they were dark. But then you again have a dark signal, and you don't know if you have background fluorescence, whether it comes from GFP or whether it's completely turned off. But GFP is turned off if you stretch it. And this is very often, even in cell culture, neglected if people interpret GFP intensities, because even in native environments, the fluorescence intensity might very well be turned off if they are part of force-bearing protein networks.
Mm-hmm. So we need some sort of other forms of microscopy, like what Guillaume was presenting, and that would somehow be able to read out tension or force or stretching, something like that. That would be have you thought of that, or any of your colleagues are working on something like this? That is not FRET-based.
This is in the moment why we developed this peptide, because we can label it with fluorophores, we can label it with radionuclei, and therefore all the conventional imaging modalities are again accessible. And that makes it then easy for the community worldwide to utilize these tools.
Wonderful. Great.
But I think we showed so far that the peptide, that we destroy this one monovalent binding motif to fibronectin. But I think we should, as a community, make a huge effort to design peptides that are acting in similar ways if they are exposed to stretched versus relaxed proteins. And that those could be, this could be done extracellularly or intracellularly as well.
We have a question from Daisy Cayley, a question that I don't quite understand, but I hope you do. As a nanoscientist, all these terms and acronyms are very difficult to me, but he or she or they is asking, is that true for all the FPs, RFPs? That means fluorescence off.
I think Darcy Kelly, who's a colleague of mine from Colombia, hi Darcy, means, is it true that the fluorescence is off if you stretch the fluorescent protein? Is it true, not just for GFP, but for all fluorescent proteins?
Correct, correct. Because you start, they are all encased in a beta barrel. Shelter. Yeah. And then the fluorescence is turned off if you change the protonation of the fluorophore. And therefore, as soon as you start peeling off, and they have slightly different mechanical stabilities, but as soon as you start peeling out the first beta strand, you already turn off the fluorescence.
Yeah, yeah. So Viola, here's a crazy idea. So if instead of using like a protein, if you were to use an organic dye and change the electronic structure with the tension, you're going to get a chromatic change or changing, and that may be another way to go at it, no? So, and then you could even expect that if you're reading out the fluorescence at a particular wavelength, it could get brighter because suddenly you'll have like a shift in the electronic structure. Yeah.
So actually, there are a lot of dyes that have been utilized in this way. They undergo a cis-trans isomerization, and they have been mostly used so far to measure membrane tension, because the ability to undergo a cis-trans transition, which you initiate by light, if you press against the fluorophore, going from trans to cis is more inhibited. And with that, people could probe membrane tension changes in a large variety of different cell types.
I have a very quick question. So what's the resolution that you can achieve with these methods? How?
Essentially, we can apply even super resolution, lattice light sheet microscopy, because we, very similar to an antibody, we just use a peptide that has the same affinity like an antibody. We bind it to 4 of the type 1 modules of fibronectin. And with that, it has nanomolar affinity. But in the way of how we apply it, it's really like antibody staining. But we don't even need a secondary antibody for it. We just photolabel it with 1 fluorophore. And with that, we know 1 fluorophore equals 1 peptide.
That's excellent. Okay, Viola, so I think it's time for our next speaker. So thank you so much for your talk and for answering all of our questions. It was excellent to share this time with you. And we will see each other again in the discussion session. All right, thank you so much. So with this, we move to our next talk, which will be given by Silvia Roque. Silvia is a full professor of photomedicine at EPFL in Lausanne. And the group in her work is focused in fundamental biophotonics. And today she will be talking to us about water as a contrast agent for membrane potentials and ion fluxes in neurons. So, Silvia, whenever you want.
All right. The slides are, you can see the slides?
Yes, we can.
All right, very good. And you can hear me too. That's all right. So water is the liquid of life, right? It shapes our planet. It shapes our climate. It shapes our environment. And the human body without water is nothing. It's just a little bit of dry matter. And 60% of the human body is composed of water. And so if you think of a cell, it actually is a very complex aqueous system. And if you want to probe molecular structure at organelles or membranes in the cells, and relate it to everything that's going on in this complex system, there's basically two approaches. There is a top-down approach where you label or you do cryo-electron microscopy, and you assume that whatever you've changed to the cell is not having an impact on the things that you're looking at on a molecular level. And the other extreme is the bottom-up approach, where you take essential ingredients from the cell, like phospholipids, you put them on an air-water interface, and then you apply very detailed molecular level spectroscopy techniques to get molecular structure. And then you hope that everything that you have extracted retains the essentials and is not different from the actual complex system. And between these two things, these two approaches, there is an enormous gap. And what we've been working on for the past couple of years is to kind of sit in this gap. I really like the aspect of getting molecular level information. I also really like the aspect of visualizing everything in time and space. But I would like to keep good control over things that are going on. So we'll start with using free-floating membranes, small unilamellar vesicles, lipid droplets, giant unilamellar vesicles, and then we apply nonlinear optical techniques and spectroscopies that we develop in our lab to study what's going on at a molecular level. So the approach is to use these systems, make a very well-controlled bottom-up preparation and characterization using standard tools. And then we use high-throughput wide-field second harmonic imaging to image the larger objects. And the smaller objects, we characterize them with some frequency scattering and second harmonic scattering. And all of this gives us molecular level information. Here we focus more on nanosystems, which I'm not, even though this is the nano session, not discussing this here. We use it to study liquid water, what is the structure of water in water, and questions like this. Very fundamental all the way to the quantum level. And here we are more interested in membranes, and in particular using water as a contrast agent to do quantified biophysics. And then eventually we apply this to more complex systems such as neurons. So before I start, I want to tell you what second harmonic generation does. We've already heard Viola mention the term. In a second harmonic generation experiment or some frequency generation experiment, you combine two photons with color 1 and 2, and you sum them and you get color 3. And this has a very interesting selection rule, namely there are no coherent photons emitted from an isotropic medium or from a centrosymmetric crystal. And so the only things that give you a lot of contrast, or any contrast at all, is breaking of central symmetry or isotropy. And this you can have in 4 ways. Either because you have a polar structure in your cell, like collagen or microtubules, or maybe you have barium titanate nanoparticles. This gives you a large contrast, and as a consequence, there is a lot of imaging going on with this kind of modality. If you don't have that, and you have a very simple system like a water membrane system, you're going to have 3 other things. The first mechanism would be that any interface has symmetry breaking in the normal direction, which gives you a breakage of structure, break in the structure, so you can see structures coming from interfaces. If you have dipolar molecules such as water, you can break isotropy by putting an electrostatic field over it. And these electrostatic fields that you have at interfaces, they are really quite strong, 10 to the 6 to 10 to the 8 volts per meter. They can orient water molecules. Hydrogen bonding is another mechanism that has enough energy to orient water molecules. So in everything I'm going to discuss, these are the 3 mechanisms for generating contrast. And so second harmonic imaging in particular relies on 2 near-infrared photons that are combined into a visible one. So in our case, it's 1030 nanometer, that goes to 550. And if you're on resonance, so the sum is resonant with an energy transition, you have a lot of photons. So if you use fluorescence, fluorophores that have this energy level, you get a whole lot of enhancement. And so this is also being used to do second harmonic microscopy by many people. Now, if you're using non-resonant second harmonic generation, it means you'll have a lot less photons coming out because there's no amplification. But every molecule in an off-resonance experiment responds in the same way. And if you now have an aqueous system with many more water molecules than anything else, and the water molecules themselves are asymmetric, this means that what you're effectively probing is water. And if you do the computation or many tests, you'll find that, in fact, if you do these experiments, you actually probe interfacial water. Now, this is really nice. So you have a water probe. The only thing that's not so nice is that the non-resonant response is weak. And this means that it has been detected in the 60s for the first time, but it's never been used because it's a few background photons that nobody gives a toss about. So what we want to do is nonetheless use this because it's very interesting. It has, if we exclude this bulk term, which we don't have, then we have photons coming from the surface or photons coming from a term that reports on surface potential. So if you could measure these things, you could actually have an optical surface potential probe, which is really interesting. Surface potentials can be converted in free energies. Free energies can be converted into membrane tension fluctuations, membrane curvature fluctuations. So this allows you to quantify a lot of things if you have this, not only in biophysics, but also, for example, in electrochemistry and other fields. So with this idea in mind, we started to look at what is the equation for the number of photons that you get from a nonlinear optical process. And it scales with the energy square and the repetition rate, and it's divided by the pulse duration. So you want short pulses. And if you had to choose between the repetition rate and the energy, you want the high energy. So basically, we started looking at, 10 years ago, what is available? What can we do? If I want to have, let's say, a microjoule, I don't want to focus this in a tight spot. I want to do wide field illumination. What repetition rate can I still use to get an image without actually damaging things? And this may be counterintuitive, but if you do the calculation, you find that if you do this, you get to lower fluences and also much lower peak powers. And so the end effect is that you can improve the imaging efficiency by 3 orders of magnitude. You can even calculate this from this equation here. If you switch from a standard light source used for 2-photon fluorescence to the one that we're using. And so here is an image of our microscope, or one of the implementations of our microscope. And on the right here, you see the contrast versus the acquisition time of an image that is composed of barium titanate and nanoparticles. And so what you see is that these are the signal to noise ratios. The optical resolution is the same, but we can take images with much shorter acquisition times, all the way 5,000 times shorter. And this is really nice because now this non-resonant response is very bright, relatively speaking. Still, with fluorescence, you can do one laser shot and then you get a good image. And that is also really nice. So I'm not, again, speaking about fluorescence, but we have a bundle of applications where we're exploring to do this, because if you have one shot per image, you can do a lot of interesting dynamics. So with this imaging system, we're going to look at membrane potential imaging, water in ion channels, and water as a probe of neuronal membrane potentials and ion fluxes. So let's start with the membrane potential imaging. So here we make a model membrane system by opposing 2 lipid monolayers through a hole. And this is a well-known technique. Montel-Muller technique is called, you make a membrane that is like 6 nanometer wide, a bilayer of phospholipids. It's like 100 micron wide. You can image it here with a white light image. You see the Newton rings of the film. You can record the capacitance and the resistance, and this tells you that you have a classical bilayer membrane. This is all technology really from the 70s and the 80s. And now we image that with our microscope. So in our microscope, we have 2 beams. 2 of those near-infrared femtosecond pulses that basically hit the membrane under 90 degrees, and the second harmonic is emitted downwards. So we look at this ring from the bottom, and if you image a membrane that's made of DOPC of cholesterol with the same composition on both sides, you see exactly nothing. And that is exactly as expected because this is a centrosymmetric system. So now, if you make a bilayer with different lipids and some cholesterol, different domains, you can see all sorts of light coming from it. Now the question is, what is this? Now in our system, we have lipids and water. So it can either be from the lipids or it can be from the water. And so by detecting the spectrum, by changing the compounds, adding ions to the solution, changing the pH, changing the charge state of the lipids, we deduced that what this contrast is from, it's from water molecules that are oriented by the electrostatic field of lipids that are ionized. So what you're essentially looking at is the water in the hydrated layer of the membrane. And so now I have an intensity that counts. How am I going to get something quantified out? So here I want to introduce you to this membrane potential imaging as we do use it on the membrane. So you have here a membrane that has a charged leaflet and a neutral leaflet. There's a potential that goes into the aqueous phase. And you have 2 electrodes on either side. And now you apply bias. So the bias adds onto the initial potential. And this is again the intensity equation as I had it a few slides back. So you have the surface term and you have the potential term. And maybe if you look at this equation, it's squared here, so it's a parabola. So now I changed the bias and I integrate the counts on all the images, and then you get this graph. So on the bottom is the bias. On the y-axis we have the intensity, and we can make a nice fit with this equation here to the parabola. And there's 2 terms that we don't know. That's this chi2 term, which tells me about the small amount of asymmetry that I have from the water molecules that chemically bind to the membrane. And the other term is the initial membrane potential difference. So now you can wonder if this is this a reasonable value? Is it done correctly? Well, we can do an independent measurement with capacitance minimization, and this actually gives us 49 millivolts. So it's pretty much perfect if you compare those 2 things. Only the capacitance doesn't tell you anything about pixel-wise or temporal changes in the membrane potential. And that's the information that we would like to achieve. And so now what you can do is look at these images here. So here's one, there's another one. And you see there's a lot of variation in the intensity. And so what we can do is we can ask ourselves, okay, where does this come from? We know that the chi2 term is very, very small, and it will not influence these large intensities. So these intensity variations, they come from the membrane potential. And so with that, we can now construct a membrane potential scale here and make membrane potential maps of fluid lipid membranes. And so here you have 3 images or videos. And what is kind of interesting here is that this is a membrane that is liquid. So there's no domains in this membrane. It's just two lipids, DOPC and DOPS. They mix. And it's completely unexpected, as far as I could tell from any literature, that there are these huge membrane potential fluctuations. And so this is a really interesting observation. We see it in many, almost every, well, not almost, in every membrane that we measure and that has a charge. And one of the things that we're working on is to understand why this is and relate this to membrane biophysics. So I won't talk about this, but there are really interesting relations, also in relation to Raphael's question just now, to a membrane tension and curvature. Because what this says is that these membranes have huge free energy fluctuations. And they, of course, relate to the degrees of freedom of these systems. And also what it means is we can't describe these membranes as passive entities with a fixed potential. That's just not how it is. All right. So having observed water at membranes and being able to get the membrane potential out of this, we now want to know is, can we measure water in ion channels? So to do this, we use alamethicin. That's a peptide that you can very easily make pores from. This peptide is in a symmetric lipid membrane. So the membrane itself is also neutral, so it doesn't contribute to the response. And we put, again, a potential bias here to change the electrostatic field. And if you do that, you can, at 150 millivolts here, you can create ion flow through the ion channels. And this allows you to quantify how many ion channels you have per image. And now, of course, the question is, when I have my ion channels open and closed, do I see a difference in the second harmonic images? And that's answer is really cool because you can. Here on the blue side, you see the voltage that's on off to turn the channel off and on. And the red trace is the integrated second harmonic intensity. So what you see is I open the channel and there's an increase in the intensity. And you also have videos of this. So you turn it, you close it, it goes down. The channel opens, and it goes up in intensity. So you see that there's a lot of intensity. Now, the one thing that intrigued us was that this is a very slow process. In fact, it takes place on a second. And the ions, of course, they go in microseconds. So why is this going down so slowly? And what we found out by doing additional experiments is that this going down so slowly because the ion channel transmits ions. That's one process. And it's oriented water in channel, but also some of the ions, they like 10 to the minus 4 of the fraction, they stay on the interface. And this transport of ions onto the interface of the membrane is building up a capacitor with oriented water molecules between. And to build up the capacitor and to reduce the capacitor, that is slow surface chemistry. And that is, because this hydrated membrane layer is a congested system with, a few moles of ions in that system, they're in there. So this is slow. And that's what you're seeing. So you see the water oriented by the channel and oriented by around the mouth, let's say, of the ion channel. Now, if you look at a picture of this, so you have different intensities, you can relate those different intensities to the different amount of ion channels per pixel. And then you can examine how the second harmonic intensity goes up and down. And you see that if you have a more crowded, higher intensity area, the dynamics is slowed down. And the way we interpret this is that these ion channels are more crowded. There's then less channels per area. And the ionic concentration in these localized structures around these ion channels, they are very high. And so you get a kind of jamming because of this high charge packing. And I'm sure that all of these mechanisms are also relevant in actual systems. So now finally, I come to my last part. I don't know how much time I have left, but I assume it's 5 minutes. Is to do label-free neuroimaging, where we want to use this membrane potential mechanism to actually use water as a contrast agent for measuring membrane potentials. And it took 6 years to do this, but the results are super cool. So here you see a picture of some neurons that we have imaged. These are static images where you see the endogenous to photon fluorescence and the second harmonic of the developing neurons. I don't want to go into this in great detail, but besides what I'm going to show you about this water, these endogenous signals also show a lot of dynamics. And I think it's really interesting. These are really interesting tools for stuff like reorganization of the cytoskeleton or metabolic activity. So I think it's really cool. We did some work on this, but we never really completed it, in fact. That has to do with my collaborators retiring. So that was kind of definitive end to the research. But now what I want to do is measure membrane potential. So the membrane potential is the resting potential of the membrane. It's described by the Goldman equation. And you can measure it with a patch clamp. Here you see what you get if you change the extracellular concentration of potassium ions. You see, boo, a change in potential. This is slow. These are not action potential. This is the resting membrane potential that you observe. So first question, can we get this type of information from second harmonic imaging? So we have our neurons in our microscope. And now recording images as a function of time and integrating the images, you see this type of curve if we do the same experiment. If you make the correlation between those two experiments, you see that's pretty much excellent. And there's no reason why it shouldn't be, because the square root of intensity is proportional to the membrane potential. And so this is really nice. But the even nicer part is if we could do this experiment and extract the spatio-temporal or real-time membrane potential. And so for that, we have here two of those depolarization cycles. These are the three neurons with three cell bodies, some dendrite and a little axon here. Second harmonic image looks terrible, right? I mean, this is labeled free image, no fluorescence, looks like crap if we don't integrate it for a long time. Still, there is intensity and these photons tell us something. So what we did is take the images. And then, of course, in the images, there are more things that give us response, right? I talked about the microtubules, I talked about the membranes. So there is a response in the image that is not potential dependent. And we can actually extract it by doing smart image processing, which I don't have time to show you, but it works. And what you get now is snapshots in this case of those two time scales of the potential in the depolarization, sorry, the potentials here, potential in the depolarization and repolarization cycle. And so what this tells you is that if we agree with ion channels being responsible for membrane potential, then we can see that there's a non-uniform distribution of ion channels in both time and space. Now there's something else you can do, because if you were in school, you did physics, you know that Q is CV, the charge is equal to the capacitance times the voltage, and we have Q for every image. So basically we can make images where we relate the potassium excess ions to the membrane potential. So we can map the ion transport using the time-lapse change in charge. And so now if I put these things together, you get videos of the membrane potential and the efflux of ions. And so now finally, I just want to show you a collection of these experiment where you basically see 2 times going over this. Oh, if my video works. Yes, there it is. So this is the time lapse of the membrane potential and the ion flux in this same experiment. So we depolarize the neurons. We do it twice. The video also plays twice. And you see all these changes in the cell bodies and in the dendrites that correspond to membrane potential and ion flux changes. Now, what does it mean on a biological level? I will say I have no clue. But I think this is really interesting to explore further. Also, if you wanted to use this for more diagnostic approaches, you could think of making something like this in an endoscope. It will never beat optogenetics or fluorescence, but it is label free. And if you want to apply it to someone who doesn't want to be genetically modified, might be nice. So with that, I'd like to summarize. I hope to have shown you that with our high throughput imaging, we can image water at the membranes, use this to get membrane potential videos and other quantified things. Then we went to ion channels. Can we see water in ion channels and around the ion channels? The answer is yes. This water contrast around ion channels is sufficient. And then finally, we went to the neurons, where we use the water as a contrast agent to measure membrane potential changes on a couple of neurons on a glass cover slip. And once you have the potential, you can calculate the ion flux as well. And so I think this gives you a lot of molecular level information about living neurons. And so with that, I should, thank my team. These are really awesome people. I should thank the ERC also and the Swiss National Science Foundation. Orly is responsible for a lot of the work together with Maxime. David and Sung Hu have also helped. And Marie, the one on the neurons, she's not on this picture because she already left when we took the picture. And so Orly went ahead and started a company. It's called Oriel Photonics. If you like our technology, he can sell you anything. So with that, I'd like to stop and thank you for your attention.
Excellent. All right. We still didn't get any questions from the audience, but Rafa has one. So Rafa, you can go for it.
Sylvie, beautiful talk. I really love your work. I always thought that SHG had a lot of potential in biology, and particularly for measuring memory potential. We spent quite a lot of time over a decade ago, with Eisenthal. But and let me confess to you one thing, that in many of our data, we're using a contrast agent, a non-centrosymmetric sterile dye. But in many of our data showed these heterogeneous patches, just like the ones that you're showing you. I mean, we never published that, and it was very, as you were saying, it goes against the dogma that the membrane is isopotential and everything. It's instantly at equilibrium. But since I'm coming to EPFL, I would love to bring some of the old data and look at it together with you and your team, because maybe it's exactly the same thing. I mean, we'll have to check whether because I don't know if the bi length of our measurement is similar to yours with water, and we were looking at the sterile dye. Anyway, so I'm just I would love to sit down with you and look together at this data, because I think you maybe confirm what you're saying, no? That there are some patches of heterogeneous.
I think it goes back to the membranes. We see it. We were surprised to see it on the membranes themselves. And what you have in the membranes is that it corresponds to like 300 millivolts type of spikes, but they are very short. And what I think it comes from, it comes basically from structural changes in the lipid that are, if you go to physical chemistry, there is a theory that tells you that this stuff should be there. And I just did 1 + 1 = 2 kind of in that sense. And then if you look at it from that perspective, it's very natural that almost any interface has this type of fluctuations. The only thing that is not natural is that when we image it or when we do spectroscopy, we average large areas and over long time scales. And when you do that, everything looks, like a constant value. But I don't think it should be constant. It doesn't make any sense from a physical chemistry perspective.
So how about labeling lipids, or like cholesterol or a different type of lipid? Can you correlate the patches with the physical chemistry difference in the lipids?
Maybe. I mean, we're working on something. You were measuring the surface potential. I have an ERC synergy with Aurélien Roux, who is a surface tension expert, and he's using one of those fluorophores that Viola was mentioning, the Flipter, which has 2 of those groups that are basically sensitive to tension. Huh? And so our plan is to work with these things, and our microscope is, of course, it's this very good fluorescence microscope as well, right? So you can get the new types of information on any fluorescent system that you want to investigate, and that's something that we're going to do in the next 6 years, basically. But it would be really, really cool to discuss this with you in more detail.
Something that I've learned from this brief discussion between you two is that one should never throw out the data that doesn't understand, because who knows, maybe in 15, 20 years, right? So for the young people in the audience, I'm a theorist, so these sort of things don't happen very often to me, but anyway, super interesting. Now I want to know what comes out of your discussion. So when you visit each other at EPFL. Well, we have a couple more questions from the audience. Marek from the CFM in Donostia is asking, excellent talk, thank you. Can you comment on how second harmonic intensity changes with temperature at constant voltage?
Oh, yes. That is primarily determined by the bias screening length. So the membrane potential has the bias screening length in the equation. And that is the one thing that is temperature sensitive.
Okay. I have a brief question from myself. So when you were showing the second harmonic in neurons and showing these patches, I could see that you were measuring two different polarizations for the second harmonic generation, but I think you didn't mention anything about the polarization. So do these two polarizations reveal different information, or what?
Well, in our experiments, those depolarization refers to the potassium that you are putting in.
Oh, no, I'm talking about the polarization of light. That's what I understood, at least.
Right. So no, that gives you information mostly about the static structure. So the cytoskeleton has microtubules, the dendrites, and the axons. And the polarization allows you to say in which direction they are growing.
Excellent. Well, that's important information, or at least for somebody like me who doesn't know that much.
Oh yeah, no, there's a lot of stuff you can say, and it's just that these people in the Fresnel Institute, Sophie Brasselet and a few other people, they're very, very good at this. So that's basically their expertise, and they're doing it so good that I rather collaborate with them than do the same stuff.
Yeah. We have one last question from Quentin Coquerel, and it says, great talk, very basic question. On one of the slides, you showed that by converging two laser beams from the top, second harmonic generation was emitted in the opposite direction. Is the second harmonic emission happening in a precise direction, not diffuse way? So an optics question.
Yes. Yeah, so I don't want to bore you with all the details, but it is basically what you have for second harmonic generation is that both of those beams have an impulse, and you need to match your momentum.
Yeah.
And so this means that if I have two beams like this,
yeah,
then the momentum is matched in this direction.
All right.
And that's where it's coming out. But this is really dependent on what you're imaging. If you're doing reflection in two beams, it comes out here. If you have a small nanoparticle, it comes out like a crazy scattering pattern.
Yeah, absolutely.
I spent half my life calculating how that works.
Me too, a little bit, on the linear regime. All right, so we don't have any more questions from the audience, and I think we should move to the next talk. We're 10 minutes late. There's no problem because we had plenty of time for discussion, but we will need to cut the available time for the discussion later. But thank you so much. And with this, we move to our last talk of the session, given by Valentin Noguer. Valentin is a group leader at the University of Bordeaux, very close to us in San Sebastian, so a couple hours away. And in their group, they work on the synaptic plasticity, and they studied it with super resolution microscopy, if I understood it well. And so, Valentin, if you have your slides ready, maybe you can start sharing them. I'm sorry if I'm not pronouncing your name right. I'm pronouncing it the Spanish way, but
yeah,
Valentin, I don't know.
Valentin, yeah, Valentin in German.
As well. So, all right. So Valentin, now in French, will explain to us today how super resolution shadow imaging works in the extracellular space of the brain. So the room is yours, Valentin.
Yeah. I'm just gonna start my timer here. Okay, yeah, hello everyone. That's very nice to be part of this, and yeah, very fitting for me to be closing the nano session right before the neuro part, because I feel really on this interface for nano imaging, but really trying to do also some neuroscience alongside with it. So in my team, we are developing super resolution approaches and try to really use them to learn something new about the brain, particularly about sort of the microanatomy, the microstructure, which turns out to be very dynamic. Now, I think techniques like STED microscopy are very useful for sort of nano and anatomical imaging, because they kind of get to us to the ground truth of at least the anatomy, not the molecular ground truth. I think there we need some other techniques, but in terms of sort of the anatomy, we're almost getting there. So I think that's why it's particularly attractive for neuroscientists. But today I will talk to you about our technique that we came up with a number of years ago called super resolution shadow imaging, to look at the extracellular space of the brain. And I will tell you about sort of why this could be actually interesting, and some of the sort of the latest turns we've taken, and what kind of interesting things we can see with this technique. Can you hear me all right? Is this all good?
Absolutely, everything's perfect.
I'm not speaking, I'm in a hotel room, so I'm not trying to speak too loudly. It's late at night here in Japan right now, so that's why. Okay, I need to advance my slide. So, of course, we agreed that the brain is a supercomputer, right? It it eats up a lot of energy and deals in information, but you can also, as an anatomically minded person, compare it to a bowl of spaghetti or soba noodles, or the tangles of neurites in the human brain. I think it's hundreds of thousands of kilometers worth of axons and dendrites. But then also you can think of the brain as Rubens vase. That's how I like to see it more recently. This is this ambiguous figure where you can, depending on your perception, can see either a vase or a face, right? And so this is, I think, just like in the brain with nervous tissue, where we can focus on the cells. So in the vein of this Rubens vase analogy, we can focus on the cells and just see the cells, or focus on the cell, the spaces that surround the cells, right? The extracellular space, or the gaps that make up the in-between spaces. And of course, to really get a full picture of things and a real view of 3D tissue, brain tissue, we need to study both, obviously. Now, about the extracellular space, it's by definition this thin gap that surrounds all the brain cells. It takes up a whopping 20% of the total brain volume. That excludes the ventricles, actually. It's filled with interstitial fluid and all kinds of goodies. All the raw material to make neurons, to make proteins and so on, have to kind of be shipped through the extracellular space. All the energy at some point has to pass through it, but then of course all the signaling molecules also. So in homeopathic amounts, ATP inside of cells is energy molecule, outside it's a signaling molecule at very low concentrations, and it passes obviously through the extracellular space. Extracellular space is nanometric, or multi-scale, depending on sort of your vantage view, polymorphic, and as we are realizing, highly dynamic. Now, why would it be interesting also to look at it? There is this glymphatic system hypothesis. Proponents are like Mike and Nedergaard. You may have heard about this idea that the brain, especially the extracellular space and perivascular space, undergo some sort of circadian rhythm, and that this has important consequences with regards to the ability of the brain to clear itself of some toxic waste products that have accumulated during high activity daytime brain life. It's interesting also from a sort of clinical point of view. There is an accumulation of pathological proteins in the ECS in certain parts of the brain. In particular, Alzheimer's, the A beta accumulates outside of the cells in some places, but not in others. And also neuropsychiatric diseases are characterized by sort of hallmark deposits in the extracellular space. And then also just from a basic biophysics, basic neurophysiology point of view, sort of neurophysiology 101 point of view, I think it's very interesting to study the extracellular space, considering that the spaces are of sort of, or the length scales of some proteins, for instance, the NMDA receptor, it juts out into the extracellular space by some 30 or 40 nanometers. So it's just around the width of the extracellular space in some places. Yeah. Okay, so what are the tools that we can use so far to study the ECS, sort of the topology and sort of the 3D shape, and also the biophysical properties like diffusivity. There are these techniques called sort of point source techniques, I should say, on the left side here. Real-time iontophoresis and integrative optical imaging are the acronyms, where basically you deposit a tracer in one place. It's can be an ion or a small little molecule that diffuses, and then you measure it somewhere else. Either measure it with an ion selective electrode, or you measure it optically. Yeah? But these techniques are quite quantitative. Can be done in live tissue, but they have relatively poor spatial resolution as you average over a big chunk of brain tissue, yeah? And also provide really no structural information. Then we have electron microscopy, which gives you all the resolution that you would ever want, but clearly there's no dynamics because all live processes arrest during the physical fixation process, and it's also prone to artifacts. In fact, the extracellular space is nearly depleted gets nearly depleted after regular chemical fixation-based electron microscopy. And then we have, on the other end of the spectrum, on the resolution spectrum, MRI, magnetic resonance imaging, gives us deep access, can be done completely non-invasive, even in human subjects, but it's obviously far from cellular resolution. One voxel contains hundreds of thousands or millions of cells. So then recently, and this may be hopefully some game changers for the study of the extracellular space, we have a couple techniques based on super resolution microscopy that were co-developed in Bordeaux actually. One is based on tracking single molecules or carbon nanotubes, which have this particularity that they sort of slowly, like a reptile, snake-like fashion, once deposited into the extracellular space, injected into the extracellular space, trace out the extracellular space and move around slowly enough that you can actually image them, you can actually track them, unlike single fluorophore single organic molecules which would be moving too rapidly. So you can track individual molecules these carbon nanotubes as they go about the extracellular space, and from that you can basically reconstruct the space, the topology, as well as glean some biophysical information about sort of the chemical properties, the diffusivity of the extracellular space fluid. Yeah. So that's one technique. And then the other technique, which developed in my team a number of years ago, is based on super resolution STED microscopy, 3D STED microscopy, where we label directly the extracellular space, where we inject a dye into the extracellular space and take basically an inverse image. Yeah. So I want to talk about this some more now. So in normal fluorescence microscopy, we pride ourselves with imaging just a few cells that are brightly labeled and to get a very high contrast, right? Sometimes if you want to see relationships between cells, then of course we label more cells, but ideally we would want to label all cells to get a high throughput sort of data out of the slice of the brain. But then if you do that, you lose all contrast basically. Given that that also there's a diffraction limit, things are even more blurry. So there's this trade-off where it's either very sparse, the label, and you get a high contrast, high resolution image, or it's too dense and it's hard to see anything, especially if you add the diffraction blur to it. So we thought about tackling the problem in a sort of inverted way, where we don't label the cells, which constitute some 80% of the tissue, but rather this 20% minority fraction, the extracellular space, right? And so this effectively decouples image quality from the labeling density. So we can get very good labeling image quality, especially if we do this with 3D STED microscopy, which gives us much sharper images. And then we end up with really an unbiased way of labeling the tissue. It's not just a few cells here and there that happen to be labeled, but it's the entire tissue that's labeled. Yet we don't have the problem of a glare of not getting any contrast, because we are only labeling 20%. We're not labeling the 80%, right? Okay, so then how do we do the actual labeling? We've either, if we do it in brain slices, we just can fill the whole chamber with a dye, or inject it locally with a patch pipette. And we are electrophysiologists, so we have micromanipulators, we have patch pipette pullers and so on. So that's no problem, and we can just inject it into the tissue. Yeah, for compatibility with our STED microscope, we use green dyes that we either excite with a 1-photon process or in a 2-photon way, and we do the quenching, the fluorescence inhibition with the light around with photons around 600 nanometers. So yes, just to recall, so we're using STED microscopy. I won't go into the basics of STED microscopy. It suffices to say it's a laser scanning technique, much like confocal or 2-photon microscopy. We have an excitation spot that's paired with a de-excitation spot, but not a spot. Actually, it's a donut. And the combination of these two spots gives you then a greatly circumscribed or greatly smaller fluorescence spot. So basically the donut, the orange donut here, acts as a photophysical pinhole to reduce the size of the original excitation spot. So we have several of these microscopes in the lab, home-built microscopes, where we can achieve in tissue resolution of around 50 nanometers laterally in XY, and in the 3D configuration, some 150 or so nanometers, 200 nanometers or so. So it's not quite isotropic, but it's certainly, we can also get a reduction in spatial resolution gain along the optical axis. And then we can, so if we combine the 3D STED with this labeling approach, this inverse labeling approach, we can get images like this. We can easily recognize cells just based on their morphology. So we don't need to label them molecularly or characterize them somehow functionally, electrophysiologically. We can do just regular STED imaging, super resolution imaging of cells, but now with the benefit of seeing the anatomical surround, the context around it. So I think that's very interesting. We don't just see this yellow cell there, this YFP labeled neuron, but we can see how it hooks up to its neighbors. There's all kinds of potentially interesting spatial relationships. Can do that also for not just for neurons, but for also astrocytes. So here we have a positively labeled astrocyte with the variant of GFP. You can see how it interdigitates into the tissue, and you can so nicely zoom in here on some very fascinating anatomical structures. Yeah, again, this is live tissue. Yeah, so we can also look at dynamics. So then with this sushi technique, with this shadow imaging technique, we can quantify some very basic parameters about the extracellular space. We can measure, for instance, the extracellular space volume fraction. You can see that it's quite variable depending on actually the area where you're looking, whether you're in the cell body area or in the stratum radiatum in the hippocampus or stratum oriens. Yeah, can vary quite a bit. So it really makes no sense to talk about one volume fraction for the entire brain, because it's going to be very regionally heterogeneous. We can measure the widths really straightforward. So just do line profiles now that we have enough contrast and resolution. Yeah. And we get data, you get widths that are highly consistent with data now based on electron microscopy that was based on cryofixation, not chemical fixation, because that gives us these artifacts. And also it's consistent with the recent CNT, carbon nanotube tracking data from our colleagues. We can look at changes in the ECS as a function of some sort of activity perturbation. So we can induce some epileptic type activity in brain slices. using either electrical stimulation, strong electrical stimulation, or in this case, pharmacological stimulation. And then we can see during a bout of sort of epileptic discharges, we can see a real contraction of the extracellular space, and then also we can see how it relaxes. We can also look at use some more subtle perturbations. We can do we can stimulate single synapses repetitively. We know that this induces structural plasticity in, for instance, in dendritic spines. That leads to sort of an enlargement of the spine head here. And now with this technique, we can also look at sort of the space that surrounds the spine head, right? And when we do that with this technique called 2-photon glutamate uncaging, we can see that things are really dynamic. When we repetitively stimulate a synapse in this way, we can see that there's a lot of sort of convoluted things happening right around the synapse, right? As you might expect, because the head is enlarging, and then other structures may be receding to make space for that, right? Then we can see also, when we look closer, we can see with very high resolution on a really good day, we can also discern synaptic clefts. There's a couple examples here. And this is, I think, due to the fact, even though the synaptic clefts are really should be out of reach for STED, because they have widths of some on the order of 20 nanometers or so. But with the contrast conditions, where you have a lot of dye in a small region, sandwiched by two pitch black structures, you have a chance of actually detecting it. Yeah. So the actual measurements are off. This synaptic cleft is not 100 or 47 nanometers or 106 nanometers. That's really reflects just the resolution of our system here. But it's, I think, interesting to see this space here, right? The synaptic cleft space. It also allows us to do some more high throughput analysis of unlabeled synaptic structures. So here we have labeled postsynaptic spines being touched by presynaptic boutons, yeah, and then we can do some correlative analysis here, and looking at, for instance, a bouton volume against the spine head volume that is from the spine head that's touching that particular bouton. Right? Quickly, should go a bit faster here. We can also use the technique to do some crude sort of connectomic analysis. Yeah, this is just some quick and dirty reconstructions based on MHJ, and very rapidly we can easily reconstruct some 50, 60, 70% of the tissue here. This is a really poor man's way of doing it. There's definitely now some more sophisticated ways of doing it. But nonetheless, I think it's an interesting perspective to do really sort of connectomic type of work with light microscopy, based on super resolution microscopy. Some other examples here: completely unlabeled cells. So it's just the labeled tissue here, and we can generate reconstructions like this, yeah, just off the bat. Another interesting application of this sushi technique is time-lapse imaging, yeah, where it becomes very nice to see sort of dynamic processes that are maybe happening relatively rarely, or where you don't know which cell is sort of you need to look at. So it's because it's an unbiased way of labeling. We can just see the entire scene, monitor the entire scene, and sometimes we see some really interesting phenomena, like in this case, a microglia cell kind of pushing itself through the tissue. Yeah. Another example shown over here, where we induce a lesion, damage the tissue purposefully here in the center, and then we just wait here. This the time axis here is in minutes, and we see several cells arriving on the scene, putative microglia cells, and they were again, they're unlabeled. They are phagocytic, as they're supposed to be. They've taken up some dye, so there's some black spots that you can see inside of them. I should go a bit faster, or just get to some newer things. So Also, the technique proves to be quite useful just in general as an anatomical imaging technique. So here we have an example where we image growth cones, which are notoriously hard to label with just GFP. It's hard to get enough GFP into the tips of the growth cones. Also, growth cones are sort of very light shy. They don't like to if you shine a lot of light on it, or if you generate a lot of fluorescence inside of them. Yeah. So in this inverse way, we sidestep that problem. Yeah, we don't need to label the growth cones, the fine tips, but we get a very good contrast here. And then we could combine this also with other types of super resolution techniques. In this case, palm microscopy, where we can locate, localize proteins, postsynaptic proteins like PSD95, and now correlate that with the anatomical structure as revealed by the sushi technique. Also, we've used it in different preparations outside of the brain, in the salivary gland, to label the epithelial structure of this organ, and then could combine this with regular positive imaging, STET imaging of some macrophages, tissue macrophages invading this the salivary glands. So then, what about using the shadow imaging approach actually without super resolution? Because this may be a bit daunting to say, okay, you can do nice imaging. It's a nice neuroanatomical imaging tool, but you need to absolutely have a 3D STET microscope maybe for it. Yeah, but it turns out it depends on what you are trying to learn from what you're trying to extract from the tissue, and what kind of resolution you actually need for that. So can you do it with 2-photon microscopy? So this would obviously make it available for in vivo imaging, open up in vivo imaging. Can you use it just with regular confocal microscopy? So this would be cool, because then a lot of people have confocal microscopes. It's highly adoptable. And then also light sheet microscopy, because that would make it incredibly fast, because 2-photon and confocal microscopes are single point scanning techniques typically, and they take quite a while, whereas light sheet microscopy can be much faster. Yeah. So just show you some recent work now, unpublished work here, where we have sort of demonstrate that indeed it is possible to get, I think, very profitable images, very interesting images using the shadow imaging approach. And this one is now here. I'm showing you some examples here from in vivo work, 2-photon shadow imaging, or TUSHI, call it that way, where we label, where we inject dye into the ventricles actually, yeah, and then just do regular cranial window 2-photon imaging. Yeah, I'll just show you here a nice movie. This is a Z-stack through looking through a cranial window above the somatosensory cortex. And you can see here a lot of contrast here, a lot of interesting things, some blood vessels, but also cell bodies, and a lot of mishmash here. This is the neuropil, axons, dendrites running through. So this is not super resolution. Yeah, we can now add super resolution to it. Yeah, we are currently doing that. And then also what allows us to do is then to do 2-color imaging, regular imaging of some class of labeled cells. In this case here, glioblastoma cells. So these are tumor cells that were injected into the mouse brain and that are now spreading in the brain of this mouse, and they're proliferating. And this gives us then a view not only of these tumor cells, but also really how they hook up with the rest of the anatomy. And I think this is a very intriguing, very fascinating perspective here, yeah, to really see to really be able to see the anatomical surround here of the proliferating tumor cells. What about confocal microscopy? So we can also get some very nice images with a tweaked confocal system. Yeah, so it's not just a regular system, but it's really one where we really take care of the spherical aberrations. Just show you an example here. So this is Z-stack, and you get a lot of anatomical details. So again, it's just confocal. Yeah, so you can try this at home. and you don't need a 3D STED microscope. Of course, if you then zoom in really and want to get to the sort of synaptic level, then it's not going to give you enough resolution there, because by definition, the confocal resolution sort of stops at 200 nanometers. But I think there's a lot of other interesting things that you can learn from these types of images. Another interesting application also is shown here, where we also in brain slices again induce a lesion, and in this case we are looking at microglial cells that are not moving their cell bodies, but they are sending their processes to the area that we damaged. Yeah? And this is a well-known phenomenon. Yeah, it's been documented some 10-15 years ago in vivo already. But I think it's very interesting to be able to reveal this now, this incredible dynamics of the microglia cells vis-a-vis the surrounding cells, right? And again, this is just confocal microscopy. Yeah? So I just want to pitch this technique, and I hope more people will be using this. And if you use it, please call it Cauchy, as in confocal shadow imaging. Then finally, we've also tried out light sheet microscopy, so lattice light sheet microscopy, which comes with it's just regular diffraction limited resolution, but the lattice variant has particularly good z-axis resolution. So in this particular microscope that we have, which is a microscope that was built on our imaging platform, so it's not actually in my lab, we have a spatial resolution of around 280 nanometers in X and Y, so it's slightly worse than confocal, but around 500 nanometers in Z. Yeah? And a really good temporal resolution, some 300 Hertz frame rate. So just show you an example here. The movie runs well, where we can just acquire stacks of images now really rapidly. So more than 100 times faster than what we are able to do with the confocal or two-photon system. Yeah? And this opens up then, looking at some really fast dynamics. And I think in this, in the context of these microglial cells that I just showed you, I think this could be interesting. Also doing some calcium imaging with this calcium imaging intracellularly, but also in the extracellular space could be quite interesting. So then I need to conclude. So yes, in a way, the brain is it's like a Rubens vase, depending on how you look at it. You can look at the sort of the membranes, the intracellular side, but also I think we should take a closer look on the extracellular side of things. And the super resolution shadow imaging technique that I presented to you today here, I think is particularly well suited for that. Yeah, so it gives us a chance to observe things in a live setting. It has it can have nanoscopic resolution if you use the 3D STED variant, and you get really a panoramic view of things. It's an unbiased view of seeing brain tissue. It's not just labeling one or few cells here and there. By design, it's completely free of photo bleaching, yeah? And there's very few signs of phototoxicity because the dye or the phototoxic materials are being generated outside of the cells where they can be washed away rapidly. Then, as I just outlined at the end, explained at the end, it's also worthwhile trying just with other very well-established non-super resolution light microscopy techniques like two-photon microscopy. Mind you, our two-photon system there uses adaptive optics, where we really tweak the point spread function and really get a really high resolution. Also, I didn't go into the optical details now, but we also use a high NA silicon oil objective for this. But still, it's just regular two-photon microscopy. Then confocal microscopy can also be used for it. Mind you, there we carefully take care of also the aberrations, the optical aberrations, and then we used it we demonstrated possibility even to use relatively low resolution light sheet microscopy in brain slices, and they also possible to get very nice high resolution and contrast rich images. So we can combine this now with calcium imaging. Actually, in the ECS, I didn't show you any data on this, but we're currently developing ways measure calcium fluctuations in the extracellular space. We combine this with single particle tracking, so we've already shown, and also just with regular electrophysiology techniques like patch clamp. In the future, we really want to go and use these combinations of techniques to study the lymphatic system that I mentioned at the beginning, look at also the various the models of neurodegenerative diseases, and particularly also tumor invasion. Yeah. So with this, I want to thank you for watching, and my past and present lab members, particularly the ones here highlighted in red, who are involved in the projects that I presented to you today. Also my collaborators here and the funding sources. So thank you.
Thank you so much, Valentin. So we do have some questions. Also, I see that Dirk is there with the hand up, so I'll give you preference for being an invited speaker of the next sessions.
Thank you so much. So I think this was fascinating. You know, I have always been intrigued by the question: how are the extracellular ion concentration maintained? Right? Have you actually looked into dynamics and gradients in extracellular ion concentrations using sodium, potassium, calcium chloride sensitive fluorophores? Is this uniformly distributed, or are there actually some gradients?
Yeah, I mean, there must be gradients, some short-lived gradients, because there are things, you know, ion, there's so big ion fluxes, and some of the spaces are very small, right? Synaptic cleft is very small. I mean, in terms of calcium, we can do back of the envelope calculation during strong bouts of activity. Actually, the synaptic cleft should be depleted of calcium transiently. Of course, then calcium comes in from the perisynaptic spaces, but for a brief moment, for some, it could be a few milliseconds, that there's some very steep gradients. Yeah. So, but we haven't. That's just a back of the envelope calculation. We haven't measured it. We're now using, we're developing with Oliver Griesbeck a sensor to measure fluctuation in the extracellular space. As you know, calcium is 10,000-fold more concentrated outside, so you need a different, you know, needs to have a very different affinity and so on.
I think it would be a really interesting application, because I always wondered how this is actually, how steady this is and how dynamic this could be. Thank you.
We have a question from the audience coming from Carles Bosch. It's a convoluted question, so I'll go slowly. So, hi, such an amazing talk. Thanks. Could you clarify on the field of view dimensions that can be imaged with sushi? Could a millimeter cube of tissue be addressed exclusively via ECS imaging, or is the C scattering imposing any hard limits on the specimen size? That's the first question. Then there is
okay. Yeah, the scattering. I'm trying to interpret the question. So the problem with the sushi imaging is that, you know, there's dye, is this very dense label, of course. Yeah, so it's not just one cell. It's, you know, up against a black background. There's dye everywhere. So that's where if it helps to do 2-photon excitation, so to limit the fluorescence to just your focal spot, right? And then you don't worry about, then you make the fact that it's not sparse, so that allows you to go deep, and you don't worry about scattering and so on, because you just have it in your focal spot. And then but then you have just 2 photons, so you should add the STED, you know, wrap around the STED donut or bottle beam to shrink down and get a tighter resolution. Yeah, so then you can go, but then the STED, you can't go really super deep, because there's the STED beam degrades as you go in because of scattering, because of aberrations. So you get a depth penetration, maybe 50 microns or so, you know, on a really good day, maybe up to 100 microns or so, but that's really the most.
All right, and then he has a second question, which is simpler. He says, related, I have missed it. Is it compatible with in vivo imaging, or is it restricted to flat specimens, ex vivo slice imaging?
Yes, definitely in vivo. So acute slices, organotypic slices, cell cultures. We've demonstrated that, and now at the end, I showed some stuff on in vivo, so cranial window, and there is just a problem with the in vivo. It's just you have brain motion, yeah, and so on, and then also issues with imaging. With the objective that we have, you have to image through a cover slip, and then the cover slip has to be really orthogonal to the optical axis, and so on. So it's a bit more pesky, but a priori, yes, you can do it also in vivo, yes.
Excellent. And Rafa?
Hi, Valentin.
Hello.
So imaginative. I cannot believe that you're going to get away calling something tushy. Yes, you know what it means in Yiddish, no? Yes. Anyway, so here's the question: have you looked at this? Whether the synaptic cleft changes in size after LTP or after learning? I mean, you
yeah,
can imagine,
we want to look at.
You have the perfect technique for
yes.
structural changes related to learning.
Yeah, it's just that's on the top of our list to do that. Yeah, in an organotypic brain slice, so where we really have the highest resolution and really can control things, and we really can stick our electrodes pre and post. It's just a time, manpower, COVID, whatever question. Getting there finally.
And I was also in some of the beautiful movies that you showed, the cell bodies were really very tightly packed. And I was surprised at how much order there is in the middle of the brain. And I wonder who's keeping all of those cell bodies in place. So do you see jiggling and people going around their business, or is everyone? So this is related to the mechanical aspects of brain function. No, there has to be, if there's so much order and precision in the way that the neurons are stacked really beautifully next to each other, someone is probably there's a reason for that, no, a mechanism for that.
Yeah, I mean, they have there's all these adhesive forces, but then at the same time, also it's very, very fluid. It's not so packed like the EM makes us believe, right? Where it's really like, super compacted. But this is after, the brain tissue gets dehydrated, then it looks artificially packed. So it's actually quite also fluid, and then cells can move through. These microglia have really permission to just go straight through it without disrupting everything. So it's a both. It's sort of stability, yet also very, very dynamic and flexible. Yeah.
Excellent. So I think this question is pretty quick, and then I think we should move to the panel discussion. Otherwise, the panel discussion will be limited to 5 minutes. So Philippe Beliki is asking, how quickly is the sushi dye cleared by diffusion when doing vivo imaging?
Yeah. So so it's from the extracellular, so in brain slices from the extracellular space, it's cleared within tens of minutes, very quickly. But then all cells are sort of phagocytic, endocytic. Some of it ends up in the cells, and then that stays there forever. Actually, in vivo, we've seen it takes around 2 days for it to completely be cleared. Yeah. So once it goes into the cells, then it's sort of vesicular inside the cells, but then also after a while that gets cleared. But the stuff just in the extracellular space that doesn't get taken up by cells gets cleared within tens of minutes. That's really very fast.
Excellent. So I think it's time to move to the last section of this session, which is the panel discussion. And in the panel discussion, in principle, we would chat all the speakers of the session together with Rafa and with myself. But I see that there are some other speakers from the following session. I don't know if they want to jump in and participate. I mean, that would be excellent. The more the merrier. So I see some cameras are turning on. We have some pending questions from all the sessions. And so, is it all of us? Sylvie, are you there? Okay. The enterprise is showing up. Okay. Excellent. So we saw a very diverse, I don't know, range of topics. So I don't really know from where to start.
Actually, you know, one thing that everyone was showing is microscopy imaging.
That's true. That's true.
It's really fascinating. We could have just had the same speakers and changed the name of the meeting. Advanced Biological Imaging 101. So it's really that shows you sort of the power of light, no? Of how you can use light to interrogate these very small structures in biology. No? Yeah, isn't it funny how everyone was presenting essentially microscopy?
Yeah,
microscopy talks.
Absolutely true.
Yeah, I mean that's just photons are just. I mean you get space and time both at a distance, and now even with super resolution. Yeah, but of course we don't really. I mean super resolution means different things. I mean we're not in the nano range, right? This is for molecular type imaging, where I think we're still far away in vivo settings. Yeah.
It's amazing also how you can see directly in all these talks how a new technique lets you discover new things and opens your mind as to new ways of thinking how the body works or the brain works. So just I mean, every one of you that spoke had examples of this in your talks. But one thing that I wanted to ask Guillaume is, Guillaume, have you ever seen, has anyone put with your thermal imaging, are there changes in temperature in cells or in the tissue when the tissue's alive? I mean, this is something I never thought of, no, that maybe there's like one part of a neuron that's hot, literally hot, and another part that's cooler. Have you noticed anything interesting there?
Well, actually, there are many works on that, many people, many research works, groups who reported some non-uniform temperature within cells in culture. And to do that, they have incorporated fluorescent molecules that are supposed to be temperature dependent. And they have seen non-uniform fluorescence. So they say the temperature is not uniform. So we actually published a paper just on that to criticize that, because according to very basic calculation, you can show that maintaining a temperature gradient at the microscale, it requires a huge power density. Because you are at a very small scale, so it's a question of surface to volume ratio. Everything cools down very fast. So I know what it costs to maintain a temperature gradient at the microscale. You have to focus a laser on gold nanoparticles, and the intensity of this laser has to be 9 orders of magnitude larger than the sunlight. And only in this case, you can maintain a temperature gradient at the microscale. So cells cannot do that. So when I could see papers showing temperature gradients at the microscale within cells, I doubt this could be true. So we published a paper with some colleagues showing that it cannot be possible. And we said maybe, yes, fluorescence should be temperature dependent, but maybe not only, and especially in a medium as complex as the interior of a living cell. And very recently, there is a group also who said our mitochondria all over our body are supposed to be at 50 degrees Celsius all the time, because they have measured a fluorescence that is, I don't know. So they published a paper on that, but now it's criticized quite a bit. So for us, it's not possible to maintain such a high temperature at such a small scale.
Can I make a comment?
You would have, sorry. Yeah, go ahead, Sylvie. Yeah, and Viola has a question too. Yeah.
I have 2 comments. One is that we did a temperature, not really a temperature measurement, but the temperature is basically the motion of molecules, right? It's kT, and so there's 2 things here. If we do a molecular level measurement of temperatures at interfaces, we can see, depending on what is if you have a constrained local structure, you have basically an effective temperature that is higher or lower. Then, if you think about it from an an entropic perspective and a perspective of just vibrational and rotational molecules, on a very confined, I don't know, nanoscale, yes, you should have local temperature, variations, considering that temperature is a thermodynamic parameter, but in the end, it reports on the degrees of freedom of molecules. So intuitively, I would say that if you have a complex system, why should you not have these differences in structure? So I'm surprised that you are. I mean, I have to look into the actual reasoning, but I would say that this should be completely normal. And on a larger scale in your body, of course, you have parts that are more warmer than others, right?
Yeah. Yeah, so at the macro scale, at the human scale, the reason why there could be a temperature increase, it's because you have billions of cells all together, like packed. And when you have billions of cells together packed, then you can have a temperature increase of a few degrees, because you have a macro scale system. But on the nanoscale, yes, you can have some variation, but I would rather say fluctuations, huge fluctuations at the nanoscale. But when you do microscopy, you average everything at the microscale. Even when you do some confocal microscopy, it's still the microscale. And since you have a dense medium, the amount of molecules and it's huge in the microtube. So you still have it, as far as you have a dense medium, you can still apply like thermal diffusion, means the normal thermodynamic law that we know. The only problem you have when you have very, very small scales, I don't think it's non-uniformities, because temperature should be, at the equilibrium, temperature should be smooth and uniform.
But I mean, if you apply thermodynamics to the body, we should all be dead.
I'm sorry?
Thermodynamically, we should all be dead. That's the lowest energy state the body can be in.
Yeah, but thermodynamics, we invented it because systems are big. When you have a very small system, it doesn't really make sense to talk about any temperature because it's fluctuating a lot. But in average, I would say the temperature is the temperature at the equilibrium of this system. If you wait long enough to have a good measurement of the temperature, it should be the temperature of, by definition, at equilibrium, the temperature is uniform.
By definition.
So no temperature signaling.
I don't think there are temperature waves in biology at the microscale. I doubt. Really.
You know, there are ways you can measure this. If you were to have a sensor for ion conductivity is very temperature sensitive, for example. You don't, it would be interesting to have another method to do that.
Mm-hmm. Yeah, but Guillaume with his method would see these things. No, I mean, you should see the mitochondria at 50 degrees if they were there. No, with your camera, no, with your method.
Well, not necessarily, because as I said, I have to subtract the effect of the cell, the distortion of the reference due to the cell, and to do that, I have to switch off my laser to have the reference image. So no, I cannot really. If they were hot, I would not see it. I would have to switch them off and then switch them on.
Viola, you have a question. Sorry to make you wait. Yeah.
No problem, because the question goes into a new direction.
Yeah, that's okay.
Yeah. Valentin, I really liked your approaches and looking at the extracellular space with your tools, and it's amazing what you can visualize. And you made one remark that that you have not seen toxicity, but then you certainly use high dye concentrations. And even if the dyes are there for 10 minutes, these cytotoxic effects should damage the cell quite rapidly, because the radicals are what makes them toxic, and the radical formation is a fast process. So how do you why do you think you see so little toxicity?
Yeah, so for one thing, it's I mean, we can now do images. We can image until the cows come home. I mean, we can really take 1,000 images at super resolution, and we don't see any like blabbing or stuff like that, which we do when we image a GFP filled neuron and do a stat on it. We can take like, 10 images, maybe 15, maybe 20, and then things go, fall apart. So that's a fact. Yeah. So I imagine what it is. The difference is that that you don't get the photoradicals trapped in a cellular compartment inside, and there's probably threshold also. At some concentrations, the cells can deal with it and compensate somehow, but then at some point it just trips the threshold, and then and this is not as easily reached outside, where also in the brain slides you perfuse, so there's a flow of solution and so on. So I think that protects them perhaps.
And then after listening to Sylvie's talk, could you essentially use non-resonant second harmonic probes to even circumvent the use of
But yeah, that's what people do actually. You have these very small. I forgot what the compound was, but you have biodegradable polymers. I guess something like polylactic acid, and you could make them into small nanospheres and use them as labels for. Yeah, I mean, I guess you need to functionalize
as diffusible labels.
Yeah, that, or you functionalize them. I mean, there are a number of groups working on this, and that gives you a high contrast. But yeah, of course, then you have to you follow those nanoparticles. What is nice about this polylactic acid is that it basically is biodegradable, and you use it as a probe for a while, and then basically it gets out of the system. At least if you're imaging, live animal or something, it goes out of the system. So yeah, that is there. I think I'm not really extremely into that field, but yeah.
Okay, so here's a question for Sylvia and Valentin. I mean, there's a lot of water in water. It's whatever. So if there was a way, instead of imaging the polarized water by the membrane, to image the non-polarized water, then someone like Valentin could go to town, no? Because you could image the whole body, no? Essentially with, is that at all possible? Or I don't know if I guess, I mean, you would have to emit some other form of water that has.
The thing is, it's everywhere, right? And so you could the only way to distinguish water from water is to do isotope dilution. That's what people do. But then you have to use infrared light.
So has anyone tried SHG of isotopic water in tissue?
Not in tissue. No, I mean I've done it, but I mean we're not getting. Yeah, it's difficult to you have a lot of background. It's difficult to.
And it won't stay outside, Rafa, right? It will go just past membranes and. So
that's true. I mean water goes everywhere.
Very quickly it will.
It's in
it has to be membrane impermeant.
It's in your proteins too. I mean, we always see these protein structures with no water, but I'll say that the protein is not itself if it doesn't have 50% of water in the volume.
Yeah, no, I'm just taking Valentin's argument to the logical limit of imaging everything that's no water by imaging water.
Yeah.
Super resolution imaging of any molecule in the cell or in the body, that's no water because you image all the water.
But since the second harmonic wouldn't pick up isotropic systems, third harmonic might be a way to go if you really are interested in looking at isotropic water.
No, it is interesting because you do pick up the isotropic water. There's I'm never saying this, but there is an incoherent response from water that is basically 50% of the signal, but we usually get rid of it because that's not the water that we're interested in, but it's there as well. Yeah. I'm going to think about this question.
Okay. I was talking and I was muted. So I don't know if anyone from the attendees has any questions from the entire for the entire panel. This could be a nice chance to say something. Otherwise, I think it's time to close the session so that we can, yeah, get some coffee or breakfast or dinner or whatever each one of us needs. Okay, nobody's saying anything, so I think we can close the session and we'll see each other at what time? Rafa, do you have it?
Yes, in 25 minutes, actually.
25 minutes, 24.
Yeah, 24 minutes from now.
Okay, excellent. So see you in a bit. And thanks a lot to all the speakers of the session. You've been wonderful. So hope to see you soon again. If you want to stay for the second session, please.
And this will be it will be broadcast on YouTube later,
yeah, but
right?
you can stay in the same link. It's fine.
Yeah, there is it's been streamed in YouTube, and after the meeting, we will post except for one talk. I think we will post all the talks in YouTube. That's the plan.
Okay, yeah. Great. Yeah.
Wonderful.
See
you
you in a second.
Yeah.
See ya.
Aitzol. Nahi duzenean? Yeah.
Aurrera, Rafa.
Aurrera. Okay, so with this, go ahead in Basque. Let's get started for the second session. And again, if you're connecting anew, good evening or good afternoon or good morning, depending where you are in the world. This is the NanoNeuro meeting, and now we're going to focus on the neural aspect of the equation, mostly, not uniquely. And we're going to start with Shimon Weiss. For the speakers that are joining us, each speaker has 25 minutes for his or her talk, followed by a 5-minute Q&A. And then at the end of the session, we're going to have a keynote from Alice Ting from Stanford University, who will have a little bit more time. She will have 35 minutes for her talk and 5 minutes for Q&A. And after Alice's talk, we'll have a joint panel discussion among all the speakers and Alice, Aitzol, and myself. So with this, let's get started. So our first speaker is Shimon Weiss. He is at UCLA, and he's one of the pioneers in this field. He particularly was one of the first people to start using quantum dots and semi-quantum nanoparticles in biology and neuroscience for functional measurements. And his talk is Novel Nano-Optical Tools for Electrophysiology and Bioelectricity Investigations. Shimon, the floor is yours.
Thank you very much, Rafa and Aitzol. Great pleasure. I enjoyed very much the first session, and I'm sure I'll enjoy the next session as well. So let me just tell you a little bit of work we have been doing both at UCLA and at Bar-Ilan University over the past few years, trying to use nano materials, nano tools as new tools to study neurons, and in particular membrane potential. So what I would like to cover, I'm not sure I'll be able to cover both topics, but I'm going to talk about what I like to call pointy voltage nanosensors, or if you want single molecule or single particle voltage nanosensors. And if time allows, I will just touch upon more recent work on bacterial bioelectricity. For the first topic, this is the collaboration with quite a few groups, both my groups at UCLA and at Bar-Ilan University, CNRS, Antoine Triller and his group, Weizmann Institute, Dan Oron and his group, Jörg Enderlein in University of Göttingen, Evan Miller in Berkeley, and quite a few people and lots of funding resources. So the vision is really to be able to follow in one field of view thousands of spots where we can take voltage measurements. Think about thousands of patch clamps, simultaneous patch clamps. We want to do it at super resolution, get basically nanometer information. We want to be able to put those patch clamps in particular locations that we are interested in, meaning we want to be able to target. And think about basically non-genetic optogenetic. We want to create tools that will allow us to work on wild type, such that we can do those studies without any transfections or transgenic animals. So I'll start describing earlier work where we try to work with quantum dots and nanorods, semiconductor quantum dots and nanorods, inorganic nanomaterials as voltage sensors. And this really goes back to the work of my postdoc mentor, Daniel Shemla, and David Miller at the labs at that time, that just after the invention of molecular beam epitaxy, they applied voltages on quantum wells and basically showed that you can get Stark effect for excitons that are confined. It's going back to the very basic concept of particle in the box in quantum mechanics. This is basically valence and conduction band for excitons in a quantum well. You apply a voltage, you tilt the bands, the energy shifts, you basically get a redshift. In terms of wave function, you have overlap of the electron and hole wave function in the unperturbed well, but if you apply voltage, you basically push the wave function of electrons to one wall and the wave function for the holes for the other wall, and you get basically this energy shift. Basically, that's the quantum confined Stark effect. So the main concept is, can we use that effect to study membrane potential? This, in fact, the demonstration of Stark effect, quantum confined Stark effect in quantum dots was done already in 1997 by the group of Mujibu Wendy at MIT, and they've shown very beautifully that you could get this parabolic behavior as predicted by theory. You could measure the polarizability of single quantum dots, but that was done at cryogenic temperature. When they warmed up the cryostat to room temperature, this effect was washed away because of spectral diffusion and other reasons. So we asked the question, can we actually take such a quantum dot and try to measure action potential in neurons at room temperature? We'll have to embed this nanoparticle inside the membrane of the neuron, and we want to be able to do this on the single particle level. So the first experiment was done already in 2012, where we did basically an in vitro experiment. It's a pure physical experiment where we have an inverted microscope. We put electrodes on the cover slip and we put some quantum dot there. We apply voltage on those electrodes and we collect the photons and measure the spectrum as function of the voltage. So here are the interdigitated electrodes, and basically here we have a slit that you see few quantum dots. We put a split just to see few of them. Then we had a prism, and basically we have a single molecule spectrometer. So here you see emission from single quantum dots as function of wavelengths. In the next slide, I'm going to rotate this spectrum in 90 degrees, and here you see a checkerboard that represent the voltage modulation on the electrodes. And you can see here very nicely this spectrum now in the vertical direction, and in particular in this area, you can see that this line representing the spectrum from a single quantum dot is going up and down. That's actually the quantum confined Stark effect that we are looking for. And good news, we can see this in room temperature. You can see some blinking from the quantum dots as expected. And we can analyze this data in several different ways. The blue curve here represents the total emission. So we basically integrate the spectrum. And you can see some zigzags. That zigzag is basically the signal we are looking for. If you are looking at the peak position of the spectrum, this is shown here in green, and this is much more sensitive. You can see very nicely the spectral changes due to the voltage. So that's the Stark effect. And now we want to use it to measure membrane potential. We have done quite a few iterations in making all kinds of different structures. This is, for example, a type 2 seeded nanorods. So the synthesis of such materials is much more complicated. These apparently are the best materials. And here you can see an average spectrum for on voltage and off voltage from such a device. And you can see on a single molecule level, we get 30 nanometer shift in the spectrum. We get 36% quenching of the emission. Basically, the effect is robust, strong, and works at room temperature. We then moved into high throughput screening of libraries of such quantum dots. I won't go into the details. This is all in this paper, but basically we could screen all kinds of different quantum dots. We can measure the same type of effects on the library. And we could also characterize the time response of this effect. It basically goes down to the fluorescence lifetime of the excited state of the exciton. Here we show that it has to be faster than 3.5 microseconds. So in principle, we can use the quantum confined Stark effect in a single quantum dot to measure action potentials, which are sub-millisecond. And among all those different bandgap configurations, quantum dots or nanorods, we identified the one that worked the best and gave the maximum signal. These are basically the seeded type 2 nanorods. So we basically need to take these sensors. We have shown that in vitro, on electrodes, they have a strong signal. But in order to measure the voltage across a membrane, we'll have to embed them inside the membrane so that they can really sense the voltages across the membrane. For biological molecules, molecules that are supposed to sit in the membrane, basically membrane proteins, there are two main ways that the cell makes sure that those molecules find the membrane and fold correctly into the membrane. One is spontaneous insertion. Usually those molecules will be cationic. They will get attracted by Coulomb interactions to one of the leaflets and slowly insert and fold spontaneously into the membrane. But the more common way of the cell taking care of targeting membrane proteins is actually an active insertion using the translocon. So there is a whole machinery to put proteins inside the membranes. We want to take inorganic nanoparticles and put them into the membranes without having that machinery. That's a very difficult task. So one way of putting those molecules in, the first way we tried, is actually to take these nanorods, and the dimensions are more or less proportional. So we have here a seeded nanorod that we want to put into a membrane. And our first idea was to coat those particles with peptides, which are transmembrane peptides. So this is shown here. Those are alpha helices that basically are engineered to have, basically we take a native transmembrane alpha helices, but we modify them. The outside has to be hydrophobic in this area. In this area, it has to be hydrophilic. We are basically trying to create those nanoparticles to behave as if they are membrane proteins. So they need to be hydrophobic here, hydrophilic at the extracellular and the intercellular ends, and we need to stick those peptides very tightly into the surface of the particles, and this is done by introducing cysteines only on one side. And we have shown through cryo-EM that indeed we can put those particles into the membrane. Many of them don't go into the right orientation, but every now and then we can see good orientation. We can even model this insertion and actually get very similar behavior in the model to what we actually measure in practice. Once we are able to put such particles into the membrane, we want to show that indeed we can measure membrane potential with those particles using the quantum confined stack effect. So this was done in collaboration with the group of Adam Cohen. And here you see basically a patch clamp. We modulate the voltage with a patch, and we get a very, very noisy signal from a single particle. However, we apply a burst search objective algorithm, and here you see some areas which are highlighted in gray. When I zoom in, you can see the same zigzag that I've shown you for the in vitro experiment. We actually see this. So we get very brief times within this time trajectory that indeed the sensor works as advertised, but most of the time we get actually very, very noisy signal. And most likely this is because the particles that go in and out of the membrane are not inserted properly in the right orientation for a long period of time. If you go on a slower time scale, we have this voltage protocol that you see here, and we collect fluorescent. You can see that more or less we get that type of behavior as we expect. But the insertion is not good, and we need to get those particles inserted and stay in the membrane for a longer time. The peptide coating technology turned out to be quite demanding and difficult. I think it could work, but it needs much more investment to get this to work well. So we switched to a different technology where we actually take the nanoparticles as synthesized with ligands on the surface. Those are basically hydrophobic ligands. The particles at the end of the synthesis are in non-polar solvent. And we solubilize them in buffer by adding lipids. And we can choose all kinds of different types of lipids to get intercalated into the ligands. And hopefully those lipids might be mobile and they might be able to diffuse on the surface of the particles. So the idea is if the lipids are indeed rearranging themselves depending on the environment that they are in, we could get behavior as we wish. If you have head groups which are hydrophilic, shown here in red, head groups which are more hydrophobic, shown here in green. Maybe if you take a mixture of lipids, coat our particles in this lipid coating approach, once we introduce them next to membranes, those lipids might move around and allow the particle to get into the membrane. So the first approach of lipid coating was done with brain extract lipids. This was done in the collaboration with a group of Antoine Triller from CNRS in Paris. And I won't go into the details. We had to do quite a bit of fancy microscopy here to really pick up only the neurons that we are interested in, the neuron that we patch, and then basically find the particles that are in the membrane of that neuron and do the same type of signal analysis that I've shown you before. Here we again modulate with the patch in a periodic fashion the membrane potential, and you can see here FFT curves that indeed show signal. And again, if you do the same burst algorithm search that I described before, we can see the same type of zigzag signals that represent the quantum confined Stark effect as I've shown you before. So in principle, the inorganic nanoparticles can be made to have strong enough Stark effect signal, and they can be put into membranes. However, that insertion still is not perfect, needs a lot of work, and just problems to consider. You all know and familiar with blinking of quantum dots. Charging and ionization leads to spectral diffusion, and basically the correlation between changing the intensity as a measure of the Stark effect and the spectral shift is not good, and this is because of this random blinking. And a bigger problem is being able to put those particles into the membrane in a very stable way, such that they stay in the same orientation and insertion for a long period of time. More recently, we focused on actually having much more precise control over the composition of lipids. So we make synthetic mixture of lipids, and two ways of inserting nanoparticles into membrane. One is we make SUVs with as-synthesized quantum dots, where the SUVs has a unique composition of lipids, and we are able to put quite large particles into membranes. So this is kind of a record. People have shown in the past they can put quantum dots up to 4 nanometers in the space between two leaflets of the membrane. With this approach, we are able to go all the way to 30 nanometer diameter of quantum dots and insert them into the membranes of SUVs. And a different approach is take the particles as synthesized, make a mixture of lipids, coat them in the particles, put them in buffer, and then introduce them to SUVs after fact. And with this approach as well, we are able to put those particles into membranes. So this is basically work in progress, and we hope that we'll be able to improve the insertion of those sensors into membranes such that we can record for a long period of time. Meanwhile, we try to come up with other pointy sensors. So one approach is very simple. We take polystyrene beads filled with fluorophores. And here we use a trick that many have used in the past before us, which is using a quencher, an anionic quencher that is inserted into the membrane. It's a hydrophobic anionic quencher, DPA. And basically, as the membrane potential changes, this DPA is moving up and down in the membrane. You are able to put fluorescent bead in close proximity to the membrane. The quenching of some of the fluorophores in the bead will hopefully give us a signal. So as we change the potential, basically we'll have more quenching or less quenching of that fluorescent bead. One issue to consider when we use DPA, DPA is in equilibrium both in outside of the cell and inside the membrane that could quench quite a bit of the bead, but hopefully there are enough fluorophores that will be sensitive to the DPA inside the membrane. So here are examples. This is a patch of a hex cell, and you see here many beads on the membrane, and we have a protocol of voltage where we modulate the voltage of the membrane and we collect the fluorescence from the beads. This is an ensemble measurement, but we can do this on the single bead level as well. So here we test how fast we can modulate and record fluorescence from the beads. You can see that as we go faster and faster, we lose signal. So this is not good enough to measure action potential, but at 5 milliseconds, we still get signals. And here you see actually single beads on neurons. So we patch the neuron, we apply at this point a different protocol, and we can collect signals from individual beads here. And we can calibrate and get calibration curves and so on. And the last nanosensor that we have developed is based on nanodiscs. You're all familiar with nanodiscs that were used to crystallize and solve the structure of membrane proteins. Basically, it's nanodiscs are patches of lipid that are packed together with a scaffold protein, alpha helical belt that holds the lipids together. And we, instead of using basically inorganic nanoparticles or beads, the question is, can we use those nanodiscs as pointy nanosensors? So the idea again will be to use DPA as a quencher. We are going to label the nanodiscs with fluorophores, attach the nanodiscs to the membrane, the target membrane, and basically as the DPA goes up and down, we hopefully will get quenching, and therefore we'll be able to sense from this nanodisc. Again, here there is issue of DPA in solution. So we, in order to improve the response of the sensor and not having the fluorophores in the nanodisc quenched by DPA in solution, we came up with two different combination of two different solutions. One is we are adding another fluorophore to the nanodisc, which take the emission far to the red, so we increase the Stokes shift. And the other one, we protect the top of the nanodisc with streptavidin. And by using those two tricks, we were able to actually develop a very nice sensor. And here again, first you see a patch clamp with HEC 293 cells, and you see the protocol and you see the measurement. This is actually, that's a mistake. This is not single, this is an ensemble measurement. And here you see a measurement on a single particle. So these are single nanodiscs on the membrane, in this case of neurons. This is a voltage protocol, and you can see that we can record from single spots. And we can even conjugate to the scaffold protein that has the cysteine at the end, an antibody, and then target those nanodiscs. In this case, we are targeting to GABA-A receptor. We have transfection of gafrin, M-cherry, and you can see very nice localization of our sensors to gafrin. So basically, we are looking into synapses. This is synapses on the main body of the cell, on the soma, but nonetheless, we are able to target synapses with this type of sensor. And here you see the recording from the targeted synapse. Again, the time response here is not sufficient for action potential, but it's a single recording from a single synapse from a single nanodisc. So what I've shown you is how modular and precise the self-assembly of those nanodiscs are. We have lots of control by having cysteines on the scaffold proteins. We can control the lipid composition of the nanodisc. We use cationic lipids to basically bind electrostatically to the membrane. And we also add some cholesterol. We use two different dyes as donor and second acceptor. We protect the nanodisc with avidin, and we can conjugate antibodies. So this technology, I think, is very, very promising. It allows a lot, as DNA origami became a major tool for nanotechnology in terms of nucleic acid manipulation. I think that nanodiscs are also fantastic nanoparticles that can be a platform to build all kinds of different functions. And we have demonstrated basically voltage sensing with these nanodiscs. So to conclude this first part of my talk, the point is single nanosensors. We demonstrated recordings from quantum dots and nanorods using the quantum confined Stark effect. We have shown a recording from beads and nanodiscs. And further improvement, we'll try to get much faster time response, such that we can get 1 kilohertz response to measure action potentials, 10 millivolt voltage sensitivity with high signal to noise from single points. And basically, the vision and our dream would be to target 2 sensors to the presynaptic and postsynaptic membrane of the same synapse, and basically record electrical activity across one synapse. If I have just a few more minutes, I want to touch on a more recent work where we collaborate with Evan Miller in Berkeley and Eduardo Charbon in EPFL to develop a technology to look not at action potentials, but actually to look at resting membrane potential. In this particular demonstration, I'll show you work that we are doing in bacteria. However, this technology could work for any tissue as well. So bacterial membrane potential plays a very important role in bacterial physiology, ATPase synthase, and cell division, and signaling, and antibiotic uptake, and so on. Most of the work that have been done to measure bioelectricity in bacteria has been so far done with Nereistian dyes, but those are very difficult to work with. It's very hard to calibrate and actually figure out what is the exact resting potential that you're trying to measure. So we want to develop more quantitative tools. And for this, we are using a set of dyes that were developed in the Miller lab. These are lifetime-based voltage sensors. One of them is, you see here, VF2.1CL. These are basically molecular wires with electron donning group at the end, which sits inside the membrane. It intercalates into the membrane. And basically, depending on the membrane potential, you have more quenching or less quenching of the fluorophore. Think about tunneling to a barrier, and you change the barrier height. So Evan already calibrated these dyes and have shown very nicely that you can measure through lifetime the voltage, the membrane voltage. And our idea is to use that to measure resting potentials in bacteria, so we can indeed stain. Here you see a field of Bacillus subtilis that is stained with this membrane, stain. And instead of actually using film microscopy, conventional film microscopy that is usually require confocal microscope with a SPAD detector and TCS/PC electronics, we are using something that is called phasor analysis. And this really allows you to save the need for acquiring enough photo rivals and building a histogram and then fitting it to sum of exponents. With the phasor analysis, you actually can do film on the fly. So the experiment that we are interested in is to have bacterial culture and then change some conditions with ionophores and basically measure the fluorescence lifetime through the phasor analysis for the 2 different conditions. So this is shown here. You see here basically Bacillus subtilis, where we change the different resting membrane potentials by using different concentration of ions and ionophores. And you can see that we can very nicely shift the resting potential of this bacteria and measure basically the mean lifetime and hence the mean resting potential for each one of these populations. And we also see some variance within populations. In particular, in this case, we see a very large variance, which is actually very interesting. And that was done, I forgot to mention that this work was done with a confocal film, but analyzed through phasor analysis. And with the help of Eduardo, we are working on a spud array camera, which is a lifetime camera. It has a gate that we can shift, and from there we can extract the lifetime. My colleague Xavier Michelet worked out a phasor analysis for this time-gated spud array camera. And here you see very initial experiments on bacteria where we can actually see the stain of the membrane and extract lifetime. And we hope to be able to look at biofilms, challenge them with all kinds of different challenges, and see electrical signaling within this biofilm. So to conclude this second part of my talk, this is work in progress. Combination of VF dyes from the Evan Miller group with the Faber film is very powerful. And in particular, if you add to that this film camera, we are basically at video rate measuring electrical signals over a very large field of view. So we hope to be able to find what is the role of resting potentials in biofilms and other non-excitable tissues. Let me just conclude by acknowledging everybody who worked on the first topic, and on the second topic, and funding, and for your attention. Thank you.
Thank you, Shimon. While we wait for questions, I have a quick one. Did you try electrostatic binding of the quantum dots to the membrane just by their, is that something that could work?
No, so there are some attempts. There is a group at NREL that have been going in that direction. We are more interested in utilizing the quantum confined Stark effect, and that really requires of putting the particle inside the membrane. There are other ways. You can do quenching, you can do electron transfer, you can do other physical phenomena, sensors, and the quantum dots, for example, for electron transfer doesn't have to be inside the membrane, it can be outside. In fact, what I've shown for the VF dyes, the dyes that Evan Miller developed, can be done for quantum dots as well. So you can have a molecular wire with a donating electron group going into the membrane, but the quantum dot can sit outside. And as the voltage changes, you will do more quenching or less quenching. So there are several different ways of developing sensors from quantum dots.
But shouldn't if you have the quantum dot in the Debye length, it doesn't have to be inside the membrane as long as it's within the electric field. No, you can still pick it up, no?
Well, yeah, but you don't want ions to screen. So the question, if you are, the Debye length, the Debye layer is very, very thin, right?
Yeah. So you worry more about the ions screening the quantum dot. Yeah. I told you have a question, Gottfried.
Yeah, so very much related to that. I've been seeing in the recent past some other approaches to measure the membrane potential using materials such as P-dot and using light. And all the proposals that I've seen are also not inside the membrane, but on top of the membrane, right? So what do you think of that approach?
That we chose particularly difficult problems.
That's totally true.
I think that, well, it's not clear to me if our approach will ever become a real technology. We have shown, basically, we validated the approach and we have shown feasibility, but we have not really taken it far enough to become a real technology.
Yeah.
It will take much more effort, more money, more time, more people working on this. I think it can be done. I think it's very attractive and very interesting. In general, making inorganic nanoparticles some kind of artificial membrane proteins, I think, is much more than just electrophysiology. You can interface cells with these nanoparticles and build in all kinds of different functions. So being able to put inorganic nanoparticles into membrane, I think, is an important challenge. What would be the best pointy sensors? At this point in our hands, I would choose nanodiscs over everything else because we have the terrific success with that. But I'm not abandoning the idea of using quantum dots to do that.
Also, do you know of any works who tried to exploit both plasmonic nanoparticles and nanodots to
Yes, actually,
quantum dots? Sorry.
So Pancho Benzania already quite a few years ago did some beautiful work, and I think Rafa asked before something about the temperature being able to activate action potential. This is exactly what Pancho did. So he basically, he excited, you know, plasmonic nanoparticles with local heating and generated action potential using this.
No, but I meant more for the reading part.
Yeah, what I want to say that in general, I see this NanoNeuro going into what I would call non-genetic optogenetics, both for reading and for writing.
Yeah. We have a question, Rafa. Do you wanna?
Yes, here's a question from Anne Escobar. Hello, thanks for the talk, Shimon. I was wondering, in the case of cold nanorods covered with lipids, if you have any idea about how long they will remain in the membrane of the cells, or if they will be internalized.
So you are asking about gold, but we have not done gold.
Yeah.
We have done just quantum dots and nanorods. We have not seen any significant uptake within 1 hour, but the particle doesn't go and sit in the membrane for a stable long duration. It probably goes in and out or change orientation, or we are not sure. It's very hard to characterize this. But when we search for this zigzag that I've shown, we can see, you know, short period of time where it is there and we can get signal. This has to be improved. So one of the ideas is to actually anchor the particle. Once it's inside the membrane, we can somehow anchor it to a membrane protein and stabilize it in there. So that's possibly an approach that will enable us to put particles and keep them very stable in the membrane for a long period of time.
Thank you, Shimon. We have to move on to the next speaker. The next speaker is Oscar Sahin from Columbia University. Oscar, if you could turn on your camera and unmute your mic. So Oscar is actually a pioneer of using atomic force microscopy in living samples, including neurons. He invented a new way to do AFM that I'm sure he will tell us about. And this links directly with the talk that we heard earlier from Viola Vogel on mechanobiology. So by developing these new tools to measure forces and tension, we can start exploring all these completely unknown side to the life of cells and neurons, which has to do with their mechanics. So with this, Oscar, the floor is yours. The title is Electromechanical Tension in Cell Membranes. Thank you.
Thank you, Rafa. And I would like to thank organizers for giving me this platform to tell a little bit about our work. So we started on this path of studying electromechanical behavior of cell membranes. After having this observation that to this day we haven't been able to explain. We work with AFM. AFM, atomic force microscope, has a sharp tip that you can use to image surfaces and their topography and their material properties. And when looking at a cultured neuron, while imaging properties of dendritic spines, and these are sub-micron sized structures where AFM is quite suitable to study, we noticed some interesting signals in our images. And what we think we're picking up is these green and red signals on the right. So the signal is the surface height. The green signal is the surface height, topography. We were seeing these little kicks. We initially called them kicks of the order of maybe 10, 20, up to 90 nanometers that last for several milliseconds. The time scale is relevant for electrical activity, but we didn't have a good reason why electrical activity of the dendritic spine would give such a large change in its height on such a short time scale. Looking a little closely into the problem, we were able to record simultaneously the force between the AFM tip and the sample. So what we were probing was the height. Now we're adding the additional channel, the force. That actually started to look like an action potential, maybe a back propagating action potential, both in its temporal features and so we thought this might be due to electrical activity, but we haven't gotten proving that. And this is actually a very difficult experiment. We see this pattern from time to time, but not always. So that proved really hard to understand. But if it is the case, it's really promising, because the signal is very, very strong, and we are outside the cell. We're not really penetrating the cell membrane. So we thought we should go take a step back and understand, study the electromechanics in a more easier to study system. And for that, we look to pioneering work by Fred Sachs's lab at SUNY Buffalo. They combined atomic force microscopy and patch clamp methods and work with Higgs cells, which are easy to work with compared to neurons. And they probed voltage-induced membrane movement and demonstrated that when you apply voltage pulses to the cell, you get changes in height. But then, and they thought this would be due to changes in membrane tension due to some electrical forces. But in the end, they concluded that this tension level is too small to have a physiological impact. We wanted to understand that, and we have a differing opinion here, and I'll tell you more later in the presentation. This is still quite a challenging experiment. We are combining an AFM and the patch clamp, essentially replicating their experimental setup. There's a cell body, you probe it with AFM, and simultaneously you have electrical contact with the cell via a patch pipette. Do the same experiment that they do, and what you see here on the screen is through the light microscope, you can see the AFM probe and the tip of the patch pipette. This bigger photo is the actual setup. And the AFM cantilever is comparable in size to a patch pipette tip, so you can see all of them together. Here's a better picture on Higgs cells, cultured Higgs cells. The cell is patched and the AFM is monitoring its height. What you see on the right side of the screen, we have a recorded displacement signal, an interaction force signal. These are coming from the AFM. And we are applying a voltage waveform to the cell. And then we monitor the AFM signals in response to that voltage signal. So we have these channels. What we essentially did in the beginning was we repeated the results, and we essentially found consistent results with the original Sachs group experiment, with the additional information of the interaction force. That really suggests that this is an electrical force that happens very quickly. The displacement is a slower response because AFM can be slow unless you really try to optimize that. But the interaction force is an open loop measurement that can be quite fast, and that shows some pattern similar to the voltage that we apply. So we can read out electrical forces through AFM, through mechanical means, and that would be proportional to the voltage across the cell membrane in some way. So that's what we're trying to understand. We also saw similar displacement patterns on neurons. So this phenomenon is not really defined by the cell type. It's a physical property of the cell membrane. So this data that I'm showing is coming from neurons. We kept studying with the Higgs cells because of the ease of the experiments. And now I would like to tell you a little bit about the physics of this. The Sachs group already pointed to something called the Lippmann effect. that could change essentially surface tension, and that they thought could be affecting the cell membrane tension through electrical means. The Lippmann effect at its heart is electrical force. So like charges repel, opposite charges attract. And if these charges are across a capacitor or on a surface, you will see the implication of these electrical forces. It's a force that is proportional to voltage squared. The forces in a capacitor can be, is basically what the energy in a capacitor is what governs this force. And the basic idea is that you apply some voltage to a water droplet sitting on a conducting surface, and the voltage that you apply can change the shape of the droplet, because the electrical forces change surface energies between the droplet and the conductor, and that causes the balance of forces at this interface, and the droplet moves. This effect is basically used in technology. In the case of the cells, you really don't have this kind of a junction, but the general idea is there. There are surfaces, and when you put charge on them, there is an additional surface energy term that is voltage square, that depends on voltage square. So they thought this could explain the results. And when you do this AFM height measurements and you measure the displacement of the tip as a function of the voltage that you apply, you get a graph like this. And this is again seen also in the Sachs Labs experiment. So we're seeing essentially the same result. What you see in the middle is the reference point, so that every other data point is measured in reference to this arbitrary zero height that we set in the experiment. There's a monotonic response. So this is actually interesting because electrical forces would be proportional to the square of the voltage. Forces across a capacitor would be voltage square. It would be proportional to voltage square. It's actually interesting, and we have a different interpretation than before. Initially, the idea was that electrical forces would change the surface tension of the membrane, and that causes, that pushes the AFM tip away. We are inclined to think that the interaction between the AFM tip and the membrane is perturbing the capacitances in the system, and that could cause the observed effect. So we're thinking that the cell membrane has some adhesion to the AFM tip. When you change the voltage, you change that adhesion. And the reason we think is that the displacement signal that we see is increasing monotonically in the range that we are observing. It's not a voltage square function. And so how we see this is that we're thinking that there are these different capacitances between the cell body and the buffer, the aqueous buffer, cell body and the AFM tip. They individually have that voltage squared dependence, but with slight differences because there's an additional bilayer between the cell body and the buffer. With the AFM tip, you're interfering with that bilayer, so the capacitances are different. And when you subtract them, you can get a monotonic result. So I'll tell you a little bit more about that. But before getting, digging deeper into the mechanism, I just wanted to summarize some very basic things about the electrical force that we're dealing with here. It's true for the membranes, but any other capacitance, if you apply a voltage difference, you have charges on one side positive, the other side negative. These charges repel each other. These positive charges. So that's the orange curve. So this repulsive force, you can think of it as like pressure that's trying to expand a balloon. This is happening in 2 dimensions, and that's an important difference. In exchange, when the system is in mechanical equilibrium, something else has to compensate for that. And in the case of a membrane, the membrane can gain some tension in the opposite direction, and those things would balance. The interesting thing, however, is that if you draw a line, the dashed line here, the net force at the dashed line is actually 0, because the tension within the lipids is going to be equal and opposite in magnitude as the electrical force that the charges are repelling each other with. So the net force in the system is zero. So in principle, some external observer would not necessarily see a tension in the system, although internally there's both tension and compression. Externally, that shouldn't lead to a change in force. In the case of a cell, there is a cytoplasm, so some of that blue force could be shared by the cytoplasm, but the cell membrane is so rigid compared to the cytoplasm that majority of it is in the cell membrane. And so we thought we shouldn't be able to see this, this effect. The source of the movement needs to be different. That force balance doesn't work in the same way when the AFM tip is present, because some of that blue force that's going to balance the electrical repulsion now comes from the AFM tip. So there's net force acting on the AFM tip in this case. When you think about this interface, that is actually similar to the Lippmann slide that I showed. When there's a droplet, it wets the surface. Now we have this triple junction that wants to move depending on the voltage. So we interpret the results based on this model, and you can calculate that with simple assumptions about the capacitances and the voltage that you apply. So the electrical forces, the voltage squared force, would follow a pattern like this, and that will be felt by anything inside the membrane, lipids and the ion channels. But the difference at the tip would be, if you can calculate, would follow a monotonic pattern. That's more like our data. So from this, we think we have a better understanding of the electromechanics of the cell membrane that is more consistent with the experimental results. And this has an implication, because if this is the case, we are thinking that that electrical force that we're unable to see that's in the membrane is a strong one. It's not a negligible quantity that could potentially affect the behavior of ion channels in the cell membrane. And for that, we will take advantage of some other aspect of our setup to address that issue. AFM can intentionally create membrane tension. So if you push the AFM tip into the cell, which is something we do in this experiment all the time, you are perturbing the cell shape, and that you're trying to enlarge the cell membrane area, but the cell membrane has a very high stretch modulus. It's like the skin on our bodies. It doesn't want to change its surface area. The underlying tissue is soft, so if you try to compress something, you will get wrinkles, but the membrane surface area doesn't really change much, so there will be tension in the cell membrane when you push into the cell. So we could use that to our advantage and demonstrate that maybe there are electrical changes when we apply some mechanically induced tension to the cell in the same setup. We actually saw a potential consequence of what I just mentioned in the experiment. So when we were probing the voltage-induced membrane movement, just before the measurement, before the AFM tip engages the cell, we were applying the voltage sequence, so and recording the current in the membrane. So you can see for a range of voltages, the current values change. We don't change anything with the voltage that we apply, but when we bring the AFM into contact, you can see the current values go up. One way to interpret this is, for some reason, the conductance of the cell increases when we bring the AFM tip, when we push into the cell with the AFM tip. So we could use that to advantage. If this is due to some mechanosensitivity, and it's not a huge mechanosensitivity, it's a factor of 2 increase in conductance in this case. If there is some mechanosensitivity, we think we can basically see sensitivity to those electrical forces. And what do we expect if the electrical forces affect the conductance of the membrane in some way? And this could be due to the mechanosensitive channels, or if the lipid bilayer has some conductance, that could also be affected by this approach. What you could see is that if you think about a basic model of open and closed probabilities of ion channels, these forces change the energy of open and closed states. So you will see a Boltzmann distribution that is affected by both the tension that you apply and the voltage squared electrical tension. So you would expect that the conductance of the membrane increase with tension, but also increase with a voltage squared relationship. And this is something we can test even without the AFM. You can take any cell, apply relatively large forces, maybe slightly out of the physiological range, and see how conductance varies with voltage. So this is something we did here. I'm just showing the mean values of conductances averaged over many cells, but their variances are on the order of 10 Siemens, so it's relatively small. What we see here is a pipette voltage on the horizontal axis. You can see that we apply quite a large pipette voltage at the extremes of our measurement, 400 millivolts. But this isn't actually the voltage across the membrane, because we know that we have an access resistance, and around this range, they're roughly equal to the membrane, resistance. So the voltage we apply is within plus and minus 200 millivolts to the cell membrane. But we take this electrical circuit into account in our analysis. So what we basically did is, what if the cell had this exponentially varying conductance as a function of voltage, squared? This is actually what you would expect. This blue curve is based on conductance that is exponentially varying with voltage square. We also took into account our access, so the values will eventually saturate to some access resistance value. The key thing that we varied here is beta and the baseline conductance. Now, if our hypothesis has some truth to it, we should also see a change when we apply tension, and that is, if you bring the AFM into this cell, these conductances should proportionally go up. And that's what we saw, these purple diamonds. And when you factor in this second component here, you see conductances are actually going up proportionally, suggesting that tension and voltage square act from the same physical mechanism. So this supports the view that there is a tension that is voltage square dependent, which is hard to detect, but its impact is present in cells. Even in the case of a hex cell, that you don't really expect a particular mechanosensitive behavior, but all channels have some degree of mechanosensitivity. So that might be sufficient to see that in our data. We haven't gotten far enough to test this with maybe adding mechanosensitive channels, known mechanosensitive channels to the membrane, but we think there are some potential implications that are worthy of consideration going forward. One is that if such a tension is present, it might mean that some channels identified in the brain to be mechanosensitive might have different purposes. So because these are places where you don't really have a clear mechanical stimulus, but the voltage square is always present. So that these channels might have adapted to be sensitive to be gated by, in some sense, non-linearly through a voltage square function. The very notion of cell membrane tension might need to be revisited, in the sense that this tension that we see is not really observable externally in an easy way. For that reason, I'd like to call it the hidden tension. It's there, its effects are present in the membrane, but externally it's very hard to see. And in line with this argument, actually, so this is not really a tension, but actually it's the opposite. It's something that would wrinkle the membrane, because the electrical forces are pushing in outward direction. That's more like a reduction in tension that would make something wrinkly. And some of the most wrinkly membranes in cells, the endoplasmic reticulum and the mitochondria, they have very large electric potentials across their membranes that have the large voltage difference. So it's interesting to think from that perspective that maybe these electrical forces are already shaping the cell in some important way that facilitate maybe enlargement of their surface area. There are some known processes in the cell membrane that affect its electrical properties, like PIP2 hydrolysis, that can have significant electromechanical consequences. So with this, I'd like to conclude and acknowledge the students who work in this group, my collaborators, and our funding source. And in particular, I'd like to acknowledge the lead student, John Jones Molina, who did almost all the experiments that I presented to you today. Thank you.
Thank you, Oscar. This is absolutely fascinating. While we wait for questions, I have a quick question for you. So these calculations that you did on the effect on channels assume just a regular plain vanilla channel. But if you work with mechanosensitive channels that have been engineered by billions of years of evolution to be sensitive to tension, you probably would expect you will have a much bigger effect. No?
Yeah, so we basically expect this coefficient, that beta, which is basically voltage square sensitivity, to go up with mechanosensitive channels. This is something we
exactly. Yeah, because I mean, I bet you, I don't know, why did Sachs say that this Lippmann effect had no biological significance?
So yeah, in their model, they attributed the entire cell membrane movement to electrical forces, and they focused on the bilayers, and from that they concluded that the force needs to be small. I actually don't know how that leads to the force being small. The exact calculations aren't available, but we think all the capacitances in the problem should be included, and that the reason of the movement is not just because of the differences in the bilayers of the inner leaflet versus the outer leaflet. It's due to the capacitance with the AFM tip. So we have a different interpretation, although the underlying idea is the electrical force. Our approach is that the AFM tip perturbs the system in a way that causes a movement, and a relatively large movement. Optical measurements also show movement in cell membranes, but those are different both in magnitude to this case. For example, with AFM, we can see up to 10 nanometers of movement. With optics, it's much smaller. And keep in mind that there is another interface here. The cell is resting on a substrate. So there's a potentially different effect from the capacitance between the cell and the substrate versus cell and the buffer. So you could expect some similar effect that could still cause the cell to move even without the AFM tip being present, because there are other cells around, or there's a substrate just below the surface. So there might be other reasons why cells move, but with AFM you can see much larger movement, and I think that's because AFM tip perturbs the system in a way that makes it move.
Oscar, there's a question from the audience from Ankush Chakraborty. Great talk. Sorry if you went over this, but have you checked if pressure on the cell membranes over longer periods and not puncturing the membrane, can have any implication on the functionality of the cells? I'm especially interested in neurons.
Okay, so our experiments last maybe 1 to 2 hours, so that the AFM tip is basically pushing into the cell for a very long time. So during that process, we don't see a major change in the cell. You can certainly puncture the cell membrane if you push harder, and that there will be very obvious changes in the system, not just electrically, but also in the cell shape. For example, the cell, think of it as a pancake on the surface. It'll turn into donuts if you push it with the AFM tip. The cells try to get away from it, and you can create holes. And those donuts, we've seen those donuts can heal again and turn back to pancakes. It's fairly complicated, but if you're puncturing it, it's easy to tell from various channels.
Any question from any other of the speakers or from anyone in the audience? We can also circle back during the discussion at the end. I think we're running a little bit late. So why don't we move on to the next speaker, Gary Zablosky. He's partly responsible or fully responsible for working in Michael Shapiro's lab, the development of acoustical targeted chemogenetics, and he's going to be talking to us about non-invasive neuroengineering. No, Jersey, if I was correct in this comment, but if not, please correct me. And the floor is yours.
That's exactly correct. Thank you so much. So let me share the screen. Let's see how that works. So can we see the screen?
Yes, we can see the screen, but not. There we go. Now it's perfect. Go for it. Yes.
Sounds good. So, and am I being heard correctly? Is the sound quality okay?
Yes, the sound is perfect as well.
Great, thank you. So yeah, so I will be talking about non-invasive neuroengineering, what do I mean by that, and about how we are trying to augment the brain function on the molecular level. So let me start that my laboratory works on 3 different areas. One of them is to finding ways for non-invasive control of the brain, the other one that acts on non-invasive monitoring of the brain, and then we try to apply those innovative tools to find new methods of potentially treating the brain disorders. So let me start with a question, which is, why don't we have cures for all diseases? Is it just something that is impossible and we just should give up and stop trying? Or is it simply very expensive and every new drug costs billions to develop, takes ages to actually become available, perhaps many years, if not decades, for some slow developing disorders such as neurodegenerative disease or cancer? And most of the time when we try to do a clinical trial, it fails. So majority of the new drug candidates end up failing in the trial. And then when we compare that to the research expenditure, it's basically nowhere near where the need is. So you can take a look, for example, at National Cancer Institute, which has about $6 billion budget per year, which sounds like a lot of money until you realize that's approximately $3.50 per US taxpayers per month. And this is for a disease that 40% people will get, and many people will spend hundreds of thousands of dollars to treat. And brain disorders are not much better. And NIMH has something like $1.6 billion a year, or 93 cents per US taxpayers per month. A third of the population can be affected by those disorders. There's 200 of them, so about half a cent per month per condition. So it's going to be difficult to treat all the diseases if we have this level of research funding. And for a comparison, to see where the public's interests are, a standard subscription from Netflix is about $15.50 per month per subscriber and cures no deadly disease. So why is it so expensive? How do new therapies come about? Well, it's just really hard to make drugs. They're very complicated. There's many steps, and at each of those steps that we have to satisfy, we're losing both time and money. And because of the practicality, we have to use somewhat suboptimal process. So the first steps, because of the cost and practicality, have to really be done in vitro or in the cell culture fashion, whereas really you want something that works in a human. So we have to optimize the whole process at the beginning. We have to develop our drugs in a system that is not really that relevant to the final product. And additionally, when we do fail, which happens a lot, we have to start it anew for every new drug, try to find new drug targets, try to new leads, and try to modify them for animal and human use. And that is quite inefficient. So the consequences is that the drug development is not very efficient and is being problematic. So you can see this cartoon where somebody is suggesting we're scraping the bottom of the barrel for the new drugs. We're seeing that we have apparently reached peak pharma and there's nowhere to go but down. That we're scraping the barrel of the drug discovery because of the Ehrum's law. So the opposite of Moore's law, where we have the decreased efficiency every year per dollar spent and we're getting fewer and fewer drugs. And finally, the worst of all, the worst culprit is the central nervous system. It takes on average 8.8 years apparently to develop a new drug for CNS. And we're only 8.2% efficient at it. Only 8.2% clinical trials are successful. which is getting dangerously close to the alpha of 0.05, which is the border for significance. So why is it so hard? Well, brain is a really difficult organ to treat. It is extremely sensitive to injury. So you cannot just poke it with electrodes and try to find something, because eventually it's going to get damaged if you target too much of an area. The injury is also very impactful on people, so you have to be very careful. It is difficult to access because there's a blood-brain barrier, there's a skull, there's plenty of neuron types, and each of them can have a different function. They're highly interconnected, they change over time. Everybody has a slightly different brain, and the diseases often develop over a very long time, so it takes a long time. We have to wait to see if there's effect of the therapy. So you really should perhaps need a new set of paradigms for treating of the CNS disorders that will be more efficient. So here are some potential areas where we could attack this problem. Well, perhaps we could reduce the workload in cells, which are less relevant to something like a complex brain, and find methods of making discovery in humans safe and ethical, and perhaps a little bit faster. We also had methods of monitoring the progress of therapy quicker, so we don't have to wait, for example, to see if somebody's Alzheimer's disease is improving over 5 years, or if it stops progressing over a decade. What if instead we had some kind of markers that allow us to see this change after a month of therapy, so we know if we need to discontinue the trial or continue? And finally, if we could do this whole process just once for multiple diseases and have some kind of platform technology that allows you to have slight changes to treat different things, then that'd be great. And to some extent, if you think about it, something like deep brain stimulation is kind of this kind of platform, because the same kind of electrode could be put in different parts of the brain, and then it could be used to treat different disorders. And this was the motivation of developing acoustic target chemogenetics. So we recognized that if we wanted to treat the brain, we first have to get to the brain. So we have to have a delivery, you have to also have a spatial precision, because different parts of the brain have different functions. And we cannot just deliver a drug that will diffuse everywhere, activate multiple different circuits, because that will have side effects. And additionally, within this brain region, we have to target specific cell types. Some of them will be activatory, some of them will be inhibitory, some of them will have other functions, and we need to target the right ones to have the specific effect on the disease. And additionally, to just add extra layer of complexity, if you want to target the disease specifically, you actually have to target a molecular pathway that is responsible for the disease. So for example, activate or inhibit the part of the circuit that you want to target. So you really need all of these to have a successful brain therapy. And so far our answer has been a small molecule. So I think it's a very big job for a small molecule to do all of this functionality in just several atoms. And it has to be small and uncomplex, because it otherwise cannot get through the blood-brain barrier. So we need some different therapeutic paradigm for us. So this is where ATTACK came from, acoustically targeted chemogenetics. We decided that we can allow for the transport of molecules using focused ultrasound, and that also can provide us the spatial regional precision. We can obtain the cell type specificity using a gene delivery vector, such as adeno-associated virus. And then we can have both the temporal control and the molecular pathway control and really tunability by addition of the chemogenetics and a chemogenetic drug. So we put it all together and try to see what's going to happen. So the first component that we'll go through is the focused ultrasound blood-brain barrier opening. So this is a technique that has been around for a while and recently has become very prominent because it works really well. So what we do in focused ultrasound blood-brain barrier opening is we apply the focused ultrasound transcranially into the deep brain region. And then at the same time, we'll infuse a contrast agent that, we may need to mute somebody. There's not a background.
Someone, someone's
so,
breaking some glasses. Yeah, there we go.
I know, it got me worried. Yeah, okay, so at the same time, we infuse the contrast agent that can respond to ultrasound. And when this contrast agent is insulated, it actually becomes compressed and then relaxes. And that relaxation can put physical pressure on the blood vessels and loosen the tight junctions in the blood-brain barrier. And consequently, this barrier becomes more like a regular endothelium that can allow viruses, nanoparticles, and small molecules and proteins to get into the brain parenchyma. That has worked very well. People actually have managed to deliver AAVs in 2012. And then also in 2018, they have done this transcranial ultrasound BB opening clinical trial. And since then, several different groups have done clinical trials on focus ultrasound. There's a very prominent group in focus ultrasound BB opening at Columbia, like Fargus group, that has done a lot of work that led to it. The second component is one of the most exciting viral vectors for gene therapy. It has very small size, which is why we use it. It's about 20 nanometers in diameter. It is non-replicating. It does not integrate very well in the genome, meaning it can be saved, probably less probability of causing cancer. And it results in very long-term expression, especially in neurons that don't divide. It can be equipped with a cell-specific promoter, so we can restrict the expression to just some cell types, and we can put a gene of interest, as long as this whole package fits in about 4,800 base pairs. AAVs have been responsible for some of the recent exciting FDA approvals for the gene therapies, such as the one from Spark Therapeutics in 2017. And the final component is one that might be familiar to many of you in this crowd, which is the chemogenetic receptors. So the idea of chemogenetics is that if we have a drug that doesn't have much side effects and can be delivered effectively, for example, because it is small, and then we express a gene that will be activated by this inert drug, then you really have a way of highlighting a cell population of gene therapy to become responsive to a remote control therapeutic. And that's exactly what the chemogenetics does. This is one of the most prominent examples of the chemogenetic receptors, the designer receptor exclusively activated by designer drug or DREAD. And that has been used quite heavily in neuroscience. There's different kinds of DREADs that respond to this compound and derivatives of it, and they can result in either decreased or increased neuronal activity, depending on which kind of drug we choose. So you put it all together, and we get acoustically directed chemogenetics, where we deliver where we open the blood-brain barrier, deliver the AAVs intravenously carrying DREAD. After some time, after a couple of weeks, have expression of the DREAD in part of the brain, and that part of the brain will become responsive to the chemogenetic drug, and that will allow us to have a specific neuromodulation. So this is what the focus ultrasound BB opening looks like in real life, as opposed to just PowerPoint. You can see this white dot right here is about the size of the opening that you can expect. And if you wanted to target bigger areas, which is very useful for large animals, for example, where injection just causes a tiny ball of expression, then you can simply move the transducer, and that's very convenient. And then if you are curious how it looks in the human, so this is from somebody else's paper, we can see this white spot that you're seeing here, the square, is actually six different BB opening sites, which is very precise. And this is what is the gene expression that results. We found that if we target the hippocampus, we can get expression in this CAMK2 alpha positive cells in both the dorsal and ventral hippocampus. But the real test was coming, can we really use this expression to have some kind of neuromodulation? Could it be useful for, let's say, therapy? So we did a very simple memory task, a fear conditioning, where we expressed the DREADs using focus ultrasound, administered the CNO and saline, the chemogenetic drug in a vehicle, performed the fear conditioning, and then checked whether mice can remember what has happened to them after we had shut down the hippocampus. And we found that we have a strong behavioral effect, that we're actually able to prevent the formation of memories by shutting down the hippocampus. So that was a success. And then I mentioned that in the context of potential platform for therapy, because similarly to deep brain stimulation, chemogenetics is allowing us to control different parts of the brain with just a couple of different genes. One that is activatory, one that is inhibitor. So if we, for example, wanted to target POMC neurons at the bottom of the brain, the arcuate nucleus, then we can modulate it, activate the arcuate nucleus POMC neurons, and decrease food intake. And that could potentially be a treatment for, let's say, obesity. But if we wanted to target the same site, but different cell types, for example, using different promoter, then it could actually have an opposite effect where we induce the intake of food and we could, for example, treat anorexia. So there's two completely different disorders that target the same site, but just target different cell types with the same gene and the same drug, and they can be treated and they can be treated with two different disorders. So by making slight modifications, as long as we have a good way of delivery, of safety of these chemogenetic receptors, as long as the chemogenetic drug works well, we could just point the ultrasound to different parts of the brain and then treat very different types of disorders. We could target, for example, the epilepsy focus and treat epilepsy, or target POMC neurons to treat obesity. So because this is such a malleable technique, I'm very interested in making it more translatable and making it better. So one of the things that we're doing is developing new kind of adeno-associated viral vectors that are mutated versions that have improved intake into the brain and decreased peripheral uptake. So remember that the AVs for the focused ultrasound deopening are administered intravenously, which means they have plenty of chances to, let's say, get into the liver and cause trouble there. And what we found that if we include about 7 mutations in the capsid of the AV9, we can have a slight increase in the delivery of this virus into the brain, and also a drastic decrease in the delivery to the liver, making it more brain specific. When we quantified that, we saw that in this first round, we're seeing about 12-fold improvement in the brain versus liver specificity. And we don't really get much expression either of those viruses in other organs, like kidneys or lungs. We found that the delivery is still site-specific, so we can get the, we don't get much expression outside of the ultrasound focus. And we also found that we can fine-tune the tropism of the virus. So you can target some cell types better than the others. And for example, in this case, we improved the capability of the AV9 to target neurons as opposed to, let's say, astrocytes and microglia. And then we went a little bit farther, and we're finding in this very preliminary data that can actually perhaps even target subtypes of neurons based on the capsid as opposed to the promoter. So fitting in a dopaminergic promoter, for example, is challenging in AV because the promoter is pretty large. But we found that we have a capsid that has a tropism that has preferential transduction of the dopaminergic neurons, at least in mice. What you're seeing here is the SNCVTA dopaminergic area within the mouse brain. The other example of how we'd like to improve the delivery is trying to find if there is a way of having the multiple deliveries for AV. And we're trying to find what is the lowest amount of immunosuppression that we can do to prevent formation of neutralizing antibodies and prevent formation of the immune response against the repeated administration. And we found that if we use this UMT mouse that has an impaired formation of IgGs, so just one subtype of antibody, we can have something like an order of magnitude more of the gene delivery of the virus that has been administered into the brain. And we don't really have much of a decrease that's not significant if we do 3 administrations of this virus at 1/3 of the dose. So this is what I was talking about with the avoiding the need of having the redoing the whole process from the beginning, right? But the other question that we raised was, can we make discovery in humans and the progress monitoring faster and perhaps more ethical? So this is a project that I'm very excited about. I actually worked very hard to put the preprint out before the conference so everybody can go and see the results, where we'll be talking about non-invasive measurement of gene expression activity in the brain. Typically, when we measure gene expression using reporters that are genetically encoded, such as, let's say, GP or luciferase, we really are trying to measure the expression within the brain tissue. And that allows us to get good spatial precision, but it's actually very difficult to see into the deep brain regions and across the skull, and becomes more and more complicated when you have a large brain, or if you want to see many different regions at once. So I'm asked, what if instead we recorded the initial location of the promoter, for example, by knowing where we deliver a gene and coding it, and then made the molecules to get out of the brain into the blood, so it can be simply tested by a blood sample, right? So we will not have this issue of the tissue really interfering with our detection. We'll be able to do massively multiplex studies because there's plenty of, let's say, proteomic methods that can detect hundreds, if not thousands of molecules within a single sample. But that's something that's not very easy with, let's say, fluorescent markers that only have several colors. So this is the concept. This monitoring gene delivery with released markers of activity, or RMA. We'll express the RMAs in known brain region. We'll allow them to be released from the brain by engineering them to cross the blood-brain barrier. And then we'll simply take a blood draw and measure what are the levels in each of the corresponding brain regions in the blood. So this is how an RMA is designed. We have a secretion signal that allows it to be released from the neuron into the brain parenchyma. We have some kind of detectable protein marker, which can be anything you want. We chose luciferase because it's easy. And to quote a John F. Kennedy, we do some things because they're easy, not because they're hard, unlike the space program. And we also have the essential component of the reverse transcytosis domain, which will allow us to have a reverse transcytosis of the molecules from the brain into the blood. And this is the result, that we have a neuron that will release the RMA. The RMA will cross through the blood-brain barrier and then enter the bloodstream, mix with all the other RMAs so you can detect it. In the first example, we wanted to see if we can monitor the efficiency of gene delivery. And we injected a single site, which covers about 1% of the brain total, with the RMA encoding gene under synapsin promoter, waited a couple of weeks, collected the blood, and did the luciferase assay, and found that they have approximately 30,000-fold signal from a single injection. So that was initially a bit of surprise, but then I realized that what happens is actually each cell produces a lot of proteins that it releases, perhaps in tens of thousands. And additionally, those RMAs are engineered to stay in blood for a long time, so they accumulate. And then you really are able to accumulate a lot of these proteins from the blood for even a small number of cells, and you end up with extremely high signal levels compared to what you could expect from, let's say, fluorescence, which would be maybe several-fold signal, not several tens of thousands-fold. And that works the same in all the other brain regions we tested. And then the second question is, could we perhaps use this to measure the brain activity in specific brain regions for a blood test, right? So that was a kind of wild idea, like we make a blood test for the brain activity. And we did this experiment where we did the injection of a special circuit that we adapted that can detect CFOS, which is activated during the heightened neuronal activity. We activated neurons using chemogenetics, collected blood, and measured the blood. This is the circuit we're using. It's called the robust activity marking. We took it from a paper in 2016 from here. And the short answer is that it's basically a very sensitive circuit that can detect CFOS and produce a lot of protein. It's like an amplifier. And when we use that circuit, we found that we have something like 4 to 5-fold improvement in the CFOS levels after the chemogenetic activation for a single brain region. So something like, let's say, a CA1 injected with RMAs, or something like the striatum caudate putamen injected with a single injection, results in 4 to 5-fold improvement in the levels of the RMA that has been induced by CFOS. And this is roughly what you could expect to see if you were doing histology. So in a way, we were able to find the blood test for the brain activity. So this is where I'll finish that. Here's the acknowledgement for everybody who made this work possible, and also for most of our funding that is listed here. And I think I'm roughly on time.
Gary, this was beautiful. How about the spatial resolution of your ATAC? Sorry, how about the spatial resolution of ATAC and spatial resolution of these released markers
of activity? Yes, let's go through that. That's a good question. So, and there's a bit of extra physics involved in spatial resolution of focused ultrasound. So let me address that. So what you are seeing here is this spot looking from the top is about 1 millimeter sized, right? And if you look from the side, the size of the opening is going to be more like 1 by 4 millimeters. Now, this can be improved or decreased depending on what kind of ultrasound frequency and hardware you use. So if you have a higher frequency, your beam is going to become smaller. So there is actually a diffraction limit that defines how big the beam can be. So we actually have some data where we combine what you're seeing behind me is the targeting of ultrasound of the VTA, which is about a couple hundred microns thick. And what we've done there was we actually combined just Krylov system with the BB opening. We overlaid the big beam with the small area of the Krylov expressing neurons, and that allowed you to have a high precision. So for small animals, you can use tricks like transgenics to improve it. You can use higher frequency of the ultrasound to have a better precision, 1 by 4 millimeters. And in humans, frankly, the resolution is not much different. It's 2 by 6 millimeters in the current hardware. And that's actually smaller than what people want most of the time. So for small animals, we can use transgenics to get hundreds of microns. And for humans, I think it actually might be good enough. Yeah, and the RMA's precision is basically the same as the gene delivery. So you get the spatial precision for RMAs based on how you deliver them. So if you are delivering using focused ultrasound, you will have the focused ultrasound's precision. If you're delivering using injection, you're going to have the same injection precision as I'm going to load up something actually. You're going to have the same precision as the injection, and then you can also use the same tricks as defining the precision with the Krylov system. So we actually have a figure where we targeted one side of the VTA. I'm going to pull up to show you the best precision we obtain. So this is the expression of the RMAs within the VTA in a mouse. It's something like
could you make it just
a
little bit bigger? Can you just make it just one
I didn't
more.
Prepare for that.
Oh, just a
I'll make it a little bit bigger. There you go.
Just one. There you go. That's it.
Yeah. So you see that this is the VTA of a mouse. It's about 0.5 millimeter here, 200 microns here, maybe 1.5 millimeters here. And by expressing the cells using the flocked element, you can actually have precision of this, right? So it's really the precision for RMAs depends on how good is your gene delivery. That's what determines precision for now. I think that, well, we have an idea how to make it a single cell, but it's a couple of papers down the road. It's not even the next paper, but probably a couple of iterations. Yeah.
Excellent. All right, more questions for Gary?
Yes, there's one in the chat.
Yeah.
Oh, I don't see it. Can you read it? Oh, here we go.
Yeah, that's fine. I do. So for the RMA technology, how do you tune protein stability so that the marker lasts long enough in the blood, but also reflects recent history of brain activity? Are there any issues with andesite proteolysis of your protein marker in blood? So this is a great question from our team. So this particular iteration of RMA is tuned for sensitivity. So it is very long lasting in the blood, about 100 hours half. So you can see the rapid changes for the increase, where you can see that there is just a derivative of the concentration of the drug, but for the decrease, not so much, right? Because it lasts so long. So we actually are making a fast version of the RMAs that's going to be faster, but at the same time, it's going to have a lower sensitivity, right? So we'll have to have a bigger cell population. The way that we're trying to compensate for that is using just some of those new single molecule sensitive tools. So even though we're going to have lower levels of RMAs in the blood, because they are faster and degrade faster, we're going to use more sensitive methods, and hopefully that will compensate for it. Yeah, but that's a good question. That's right. Right now, it is best suited for the slow changes, like long-term changes in, let's say, development of disease or monitoring of the efficiency of gene delivery to the brain. Yeah. So that's great.
Excellent. Great. Okay. I don't see any questions from the audience. So why don't we move on, because there will be opportunities for more questions, to our next speaker, which is Dirk Trauner. So I have to confess to you that when we organized this meeting, we spent a lot of time trying to make a schedule where everyone would speak at an appropriate time given their time zones. And our surprise was that with this post-COVID stampede where everyone is traveling everywhere, no one is where they're supposed to be. So Shimon Vai is supposed to be in LA, but he was in Israel. Valentin was supposed to be in Bordeaux, but he's in Japan. And myself, I'm supposed to be in New York, and I'm in Spain. Anyway, so Dirk, I said, well, at least Dirk is going to be in New York. Guess what? He's not in New York. He just moved to Philadelphia. So,
same time zone.
The same time, so yes, but it's I would say we should put a freeze on people moving just for a little bit. Otherwise, it's just it's too confusing. Anyway, so Dirk is one of my favorite people in science. He's a chemist, and he's here representing an area which is optochemistry, which has a lot to say and has a lot of potential. And he's arguably the world's expert in optochemistry, the combination of light with chemical tools, and he's done phenomenal work already in biology and neuroscience. And the title of his talk, as you see here, is Controlling the Fate and Function of Neural Proteins with Proximity Photopharmacology. You can take it from here, Dirk.
Thank you very much, Raphael, for this very kind introduction and for inviting me to speak here, and for giving me an opportunity to update you on our efforts to control neuronal function with synthetic photoswitches, what we actually call photopharmacology or optopharmacology. And I'm particularly interested in talking to you today about molecular glues, photoswitches that function as molecular glues to bring factors together but also photoswitches that are tethered to the receptor of interest to control these receptors with light. So let me start out with the first topic. Like everybody else, we are fascinated with proteolysis, proteostasis mediated by ubiquitin ligases, which of course now are at the very forefront of an exciting new direction in pharmacology known as PROTACs, as proteolysis targeting chimerae, which are bifunctional molecules that bring together an E3 ligase that provides the ubiquitination with, in principle, any protein of interest. Of course, this is not our discovery. This is Craig Cruz and Ray Deshay and Chip Radner and the likes that have introduced this fascinating concept. And once this ternary complex is formed, ubiquitination of the protein of interest happens, polyubiquitination, then the whole thing falls apart, and the now tagged protein of interest gets dumped into the proteasome, so to speak, for degradation. And the beauty of these PROTACs, and why everybody in pharmacology is excited about it, is the fact that these are catalysts. They're catalysts that activate other catalysts. In other words, they can turn over, and while some of them have very large molecular weights, they still get enough into the cell due to the catalytic function to be efficacious. Now, with my background, we couldn't resist from turning PROTACs into PHOTACs, into photochemically activated PROTACs. It's admittedly a case of acronym-driven research. So the idea was that the PHOTAC would be inactive in the dark, and once we irradiate it with light, it becomes an active PROTAC, then does what PROTACs do, turns over, then it can thermally relax to the inactive form, and it can be actively switched back to the inactive form, which provides kind of a safety switch, because there are some concerns about these PROTACs that they're catalytic poisons, and with our technology, perhaps we could turn them off with light or let them thermally relax. In addition to this, of course, light provides, I don't have to tell this to this audience, excellent spatial and temporal resolution, which I think could be particularly useful in neuroscience. So let me briefly touch upon how we conceptualize or how we actually design these PHOTACs. We take existing PROTACs, initially such as this one here, which has a ligand for an E3 ligase known as cereblon, broadly expressed, especially in the brain, and then through a linker is tethered to a ligand of a protein of interest, which in this case was a PET protein, BRD234. And we squint our eyes and we see an azobenzene photoswitch. Here's the azobenzene photoswitch in the cis conformation, which is the active one, as it turns out. So the idea is to make a molecule that has a photoswitchable ligand for an E3 ligase. The photoswitch, by the way, could also be on the side of the protein of interest. It could also be in the linker. But we, for modularity reasons, decided to first make a photoswitchable ligand for an E3 ligase, which in this case, in the transform of this NN double bond of the azobenzene photoswitch is inactive. If you radiate it with violet light, in this case, it becomes cis and active. It was designed such, and then it can function as a PROTAC. I'll spare you the synthesis. As a synthetic chemist, I'm very fond of it. Suffice it to say that we start with a multi-billion-dollar drug, lenalidomide, and turn it into these PHOTACs. And suffice it to say that this is very modular because we can put all kinds of ligands for the protein of interest on the right side here. We interrogate this with our cell discos. These are 96 or in this case 24 LED arrays on top of which we put our cell culture plates, and then we do Western blots or other investigations. Here are several of these so-called cell discos in action. This whole area of my research was pioneered by Martin Reinders, whom I want to acknowledge here, a fantastic graduate student who is now a postdoc in Munich. And here are the data from our published paper. You see that in the dark, when we look at BRD3, for instance, at this epigenetic reader, BRD3, there is no effect on protein level when we expose the cells to our PHOTAC. However, if we pulse irradiate every 10 seconds for 100 milliseconds with 390 nanometers with violet light, we see beautiful degradation kicking in at about 100 nanomolar. And then at 1 micromolar, it's completely gone. At 10 micromolar, it comes back. And that is a hallmark truly of these PROTACs, because you have to form a ternary complex. And if you have too high a concentration, you saturate both sides, the ternary complex cannot be formed, which is necessary for the transfer of ubiquitin. So this is a classical hallmark known as the hook effect that our compounds also function as PROTACs. Let's look at the time course. Let's look at BRD4. So after about 6 hours, it's pretty much gone. The speed of this degradation is dependent on the target protein because it depends on resynthesis rates. We're particularly excited about this BRD4 aspect because it indirectly also controls CMYC, which is notoriously difficult to target this cancer gene directly, since it's a transcription factor which is not really folded in the absence of its target. But we can do this indirectly using the PHOTAC technology. And then at some point we push ourselves into apoptosis, as shown by a cleaved PARP1. And that we knew early on, because the first thing we always do are viability curves. This viability curve here shows that this molecule is much less toxic in the dark than upon irradiation. In the dark, it's still an inhibitor, by the way. Upon irradiation, it becomes also a degrader. And what this graph also shows is that we can do what I like to call chromodosing or color dosing, because the photostationary state between the inactive form and the active form is a function of the wavelength of light. And we can dial this in with different wavelengths of light, the strength of this effect. So we have a knob to tune the effect of this PHOTAC, so to speak. And we have done this now with a wide variety of targets, which brings me to neuroscientific targets. So we've done quite a bit with this BRD, bromodomain containing epigenetic readers. We've done FKBP12 that was published, photodegradation of FKBP12. Not published is CDK4/6, aurora kinase A, BCR-ABLs, NBM2, FAK. But today I would like to talk about KMK2 and perhaps PKM-zeta, but particularly KMK2, and also want to briefly touch upon the genetically encoded version, where we actually tag the protein of interest with a genetically encodable tag that then provides degradation. Let's focus on KMK2. I think towards this audience, I don't have to explain the importance of KMK2 in memory consolidation, in synaptic strength formation. I find it fascinating as a chemist how this expands and then becomes permanently phosphorylated and activated. And of course, there are numerous small molecules that inhibit this kinase, calcium calmodulin independent kinase 2, such as bosutinib. This is actually the crystal structure of bosutinib bound to KMK2. And we have made the corresponding PHOTAC by putting our photo-switchable ligand targeting the cereblon E3 ligase together with bosutinib. And again, this is the inactive form where the NN double bond is trans configured, whereas the active form is in the cis configured. This, of course, required some engineering and some calculations and whatnot, but I spare you the details how we came up with this. But suffice it to say that in the dark it's inactive. Upon radiation, it becomes active. By the way, this was John Korean's structure that I wanted to shout out. And it turns out that KMK2 is hard to monitor with Western blots because it's so abundant in the brain, and small changes of KMK2 in the synapse make all the difference. And therefore, you might not pick this up with Western blots, which is why we decided together with Andrew Fenton from MVU Neuroscience to use immunohistochemistry to look into local degradation of KMK2, optical degradation in the hippocampus. For this, we prepare slices. So this is now an application of KMK2 of PHOTACs in tissues. And then we irradiate or not in the presence or the absence of our compound as a control. We do tissue fixation, and then we can stain with immunohistochemistry. It turns out that this was actually done in the suprapyramidal blade of the DG. When you look at levels of KMK2 in the dark, you see that this gets not degraded in the neurons. The neurons are healthy, as evidenced by parvalbumin staining and DAPI staining. There is no effect on the health of the cells themselves. But if we pulse irradiate with the 90 nanometers, KMK2 alpha, the neuronal form subtype, is gone, largely gone. And here are the statistics. We've also done controls with artificial cerebrospinal fluid to demonstrate this, that we can locally degrade KMK2 in an area of the hippocampus, KMK2 alpha, using this PHOTAC technology. And it's not only possible to do this in a sort of XY level of precision, but we can also run a C-stack because we can adjust the intensity of the light. And then depending on how well it gets absorbed and how much light is left, we actually see that along the C direction, we see decreasing activity of our PHOTACs as we move further in over a length scale of about 25 microns. That means you can also dial in on the C direction which level of your tissue you want to affect with the photoswitchable degrader, with the PHOTAC radar. And then, of course, Andrew and I were interested in proving that synaptic strength gets affected in a light-dependent fashion with slice electrophysiology. So we do the same protocol. We incubate the slice in darkness or in the presence of light pulse irradiation again, with or without our compound. And then we measure field excitatory postsynaptic potentials, or this slope of field excitatory postsynaptic potentials as a measure for synaptic strength. This was done by Claudia Schuh, a graduate student in the laboratory of Andre Fenton at NYU Neuroscience, our collaborator. And it turns out that in the dark, which are these open circles with the single experiment, there are no, on average, there is no change in synaptic strength as measured by FEPSPs over time. Also, when we just irradiate in the absence of our compound, in the absence of our photoc, which are these blue filled circles, we see no change in synaptic strength on average. However, when we expose the slice to this light pulse protocol in the presence of our photocs, we actually see a very nice decrease in synaptic strength as measured by these FEPSPs. It actually comes back, I haven't shown this here, over time when we stop irradiating. And here are the biological replicates. So this is n equals 7, where we can see that on average there is no change in absence of our compound. With irradiation, there is no change in an average in the presence of our compound without irradiation. And if you do both, if you irradiate and our compound is present, we see a very nice change in FEPSP slope. All right, so that is an application of a diffusible photoc. However, that does not really take advantage of genetic engineering. But of course, there is a way to bring in genetics. So I would argue that this might be another dimension, another aspect of optogenetics, because we can now genetically tag our protein of interest with some motif, with some tag that binds to a ligand that we can make a part of a photoc. As for instance, the D tag, this is derived from FKPP. It's a mutant of FKPP, a bump hole mutant of FKPP, but a non-photoswitchable version was already demonstrated by Craig Cruz. Or for us, more exciting actually and more powerful at this point is the so-called A tag, which is not widely known. It was introduced by C4 Therapeutics by a company. And is small. It's actually a little bit smaller than the D tag and has simpler chemistry to target it. So we actually like the A tag. And the idea again is that you genetically target your protein of interest with a tag, and then you degrade in a light-dependent fashion to have both light and genetics as a component of precision. And it turns out that this actually works quite well. This is MTH1 itself, which is actually the progenitor of the A tag with very low concentrations, with 10 nanomolar in the presence of light. We see beautiful degradation, 100 nanomolar, it's pretty much gone, whereas in the dark, there is no effect on the levels of this protein and by extension on tagged protein. So stay tuned, we will publish on this hopefully soon, also with more relevant neuroscientific targets. But I find this potential interesting, as I said, new direction in optogenetics to tag a factor of interest genetically and then locally or time dependent degraded with light. I would like to flip over now to a second topic. How much time do I have? I hope another 10 minutes or so, 5 minutes perhaps. I would have loved to talk about optical control of membrane fluidity by feeding cells with photoswitchable lipids, which we have just posted on bioRxiv. And given the previous beautiful talks on membrane tension, I think this has a lot of potential in neuroscience, but alas, due to COVID and whatnot, we have not done much with neurons yet. But if you're interested in feeding photolipids and photolipid precursors, photoswitchable fatty acids to neurons, and then see what effect this has on not only internal membranes, such as the endoplasmatic reticulum, but also the plasma lemma, please talk to us, because I would like to push this. I would like to, however, continue briefly by pointing out that we are still very much engaged in tethered photopharmacology, where we tether a photoswitchable ligand to a protein of interest that is genetically tagged with some bioconjugation tag, a SNAP tag, a halo tag, or a clip tag, for instance. And they have some years ago, 7 years ago, already published that this works beautifully with mGlu2, with family C GPCRs, which we internally extend with a SNAP tag, and then we link molecules like this, which have a electrophile that is stable in under conditions that it hits the tag, and then a photoswitchable version of glutamate. And this is all published. This is a model what this looks like. In orange, you see the snap tag. In gray, early model of mGluR2, which since has actually been elucidated with cryo-EM. And together with Udi Isakov, a long-standing collaborator in this field, with John Flanner, we showed that it can virally transfect the retina of blind mice and then restore visual function using this tailored photopharmacology approach. So this was published already 5 years ago, but we have stayed true to this. It turns out that this not only works with family C GPCRs, this works beautifully with family A GPCRs as well, which can be N-terminally extended, genetically tagged, and then you mount on your photoswitchable ligand on a long flexible tether. Local concentration is still high. There is proximity pharmacology definitely present. We're talking of high millimolar ranges here in some cases, or at least millimolar ranges. And we have recently posted a paper in a chemistry journal together with Josh Levitz that shows that we can control 5-HT2A function with tailored photopharmacology. Again, here I don't have to explain 5-HT2A as a target of LSD and psilocin. We made a very simple photoswitchable version of serotonin of 5-HT, a long flexible tether, and then the bioconjugation handle here to be able to control receptor function as indicated here with calcium imaging upon irradiation with 380 nanometers again. This was a proof of concept study. We're going much deeper now together with Josh, who of course is a fantastic neuroscientist, to show that this works in brains and with longer wavelength, but stay tuned for this. There's a new series of X-ray and cryo-EM structures of 5-HT2A receptors that allow us to model this and to define this better. I'm actually quite interested in the fact that 5-HT2A is the only subtype of 5-HT receptor that is co-activated by lipid. And we actually think that our photoswitchable portion of our molecule might occupy this lipid binding site, but these are molecular details that we'll disclose in time. I want to finish with a more conceptual slide again, or a series of slides, by pointing out that the docking station, so to speak, of our tethered photopharmaceutical doesn't have to be the receptor mounted to the receptor itself, meaning we can work with native receptors at native expression levels, and we can transfect cells with what we call an M protein, a transmembrane protein, single pass transmembrane protein that features a SNAP, halo, or whatnot tag. Very easy to transfect with this small gene and modular. And it turns out that this provides cell specificity to the power of genetics, tissue specificity, and this works actually beautifully. Lateral diffusion is good enough, or maybe there's some clustering, that this seems to be a generally applicable concept. We call this MA portal, from membrane anchored portal. Again, we have developed this with Udi Isakov, and we've demonstrated that we can do this with D1 receptors. We have the data here. When we do some neck engineering for this docking station, we actually see activation with blue light now as large as saturating concentrations of dopamine. We can do this with D2 receptors. This paper is in the making. Very powerful halo tag linked now, MA portal, where we can show that with blue light we see again activation as with saturating dopamine concentrations. And in unpublished work, we have resumed our work on optical control of opioid receptors. Many years ago, too many years to think about, we already showed that we can make a photosoluble fentanyl, which was active in the dark and then became inactive upon irradiation. But together with Josh Levitz again and Giovanna Romano, we've recently shown that we can make a derivative of morphine that is inactive in the dark. It becomes active upon irradiation, and that can be tailored to allow for the optical control of opioid receptors. With that, I'm done. I would like to point out the students who were critical for this program. Belinda Hetzler, now in the Bay Area, made tailored photopharmaceuticals for dopamine receptors. Tom Coe and Joe Flores led the charge on the ChemK2 photodegradation, and I already mentioned Martin Reinders as a pioneer of photopharmaceuticals. And as Raphael pointed out, I'm now in deep conversations with Benjamin Franklin. I'm very excited to be at UPenn with an appointment both in medicine and in chemistry, and I really look forward to branching out and forging collaborations with the neuroscientists here on campus. Thank you.
Thank you, there. Congratulations on the appointment and congratulations on the beautiful work. What a treasure trove of chemical tools for us neuroscientists. So here's a question, relates to PROTAC, but also for the other tools you mentioned. How about 2-photon? Have you tried this with 2-photon excitation so that you can have single cell precision with these optochemical methods? Yeah.
We, saying Udi, have done it, right? We also teamed up with David Gregorio some years ago, where we made a photosoluble version of glutamate that we could turn on and off so fast that we could beat diffusion. So definitely 2-photon is an option. Azobenzines generally have a pretty good 2-photon cross-section. So if there is interest in doing this, I would be happy to develop this further. I'm personally not equipped in my laboratory to investigate 2-photon methodologies. We just don't do this. I'm not a biophysicist. I'm a synthetic chemist who dabbles in neuroscience. But if there is definitely no strategies to increase the 2-photon cross-section, and azobenzines in generally are not bad in that regard.
Yeah. So does PROTAC use azobenzines as well, or no? I missed that.
Everything we do uses azobenzene photoswitches because they're just so photostable. Azobenzines flip instead of doing intersystem crossing. So we don't deal with any photosensitization of oxygen, which is death for photoswitches. So we always come.
I mean, I can just imagine an experiment like the one that you guys have done with the Fenton lab in hippocampus with single cell precision or single spine precision. How about turning off kinase 2 in one spine? So 2-photon can do that. So you have
I would like to collaborate with you on that. Absolutely.
Wonderful. So more questions for Dirk from other speakers, anyone in the audience?
I want to briefly throw out, if anybody is interested in thermopharmacology, where a local temperature jump of 2 degrees Celsius is useful, please talk to me. We are soon coming out with a proof of concept paper on what we call thermopharmacology.
Actually, I cannot help myself to ask you a little bit more about that, since we had this wonderful discussion earlier about the thermal effects. So can you say something more about this thermal pharmacology?
We can wrap up a ligand with a polymer that is thermally responsive and that folds upon a with a temperature change as opposed to unfold. You can figure out what it is. It's polymipam, right? There's a polymer called polymipam, which actually folds up, collapses upon irradiation. And we have found chemical ways to functionalize this with ligands such as glutamate or with rectifunctional groups. And I like the idea, but I haven't thought it through in terms of potential biological applications, other than if you get a fever, suddenly the ligand becomes inactive. So we actually can do this right in the range between 35 and 37 degrees Celsius.
Excellent.
So the thermally responsive polymers have huge potential in neuroscience.
There's a question here about thermopharmacology. Can it be used in cancer from Ganendra Singh?
If you have a local increase in temperature, you can think about it. Right now, we can inactivate the ligand that way. We cannot activate the ligand, but we can inactivate. But of course, you can inactivate an inhibitor. So you can do a sign inversion, so to speak. And we can activate and inactivate. It's reversible. We can activate and inactivate covalent reactivity. So you can think about snap tagging something and then having it locally reacting with a temperature change, which of course you can also impart with some form of irradiation locally.
Excellent. Jerry, do you have a question, or does anyone else has a question for Dirk?
What is the size of the thermopharmacology polymer? How big is it? How would you deliver it?
10 kilodalton. It's very small. It's about 10 kilodalton. So it's like a small protein, like a mini protein.
Very nice.
It should be diffusible. It should be able to diffuse for tissues rather, maybe even extravasate for the BBB.
Maybe if it's okay with you, we can just turn this into the panel discussion, and then anyone can ask questions to anyone else. So this could be a big question salad. So So Shimon, if you could turn on your camera as well and just join us. Sorry. So here's a thought. Combining methods. So I mean, Jerry's talk was inspiring for me because he's really combining three methods, for example, in what he showed us at the beginning. No? So I'm wondering whether the future, I mean, we're all developing our own little method and improving that, but maybe it's could be a good time now to start sort of using several tools at once and maybe profiting from the best. Sorry, that each tool has to offer. I'm going to close the window and the motorcycles are coming. Anyway, so this is just an open question for any of you about combination of methods, whether it's is there opportunities that are not being exploited because we're sort of in our own little niches. Yep.
Well, yeah, I think the question that Jerry asked me about, incorporating ultrasound gating or, magnetic field gating, into the types of tools we're using is very much, in the direction of, spanning different fields to bring in the best of both. Yeah, so that's very much worth doing. And, I should go hang out with Jerry and we should brainstorm about these kinds of things.
Yeah. Well, I'm asking this question because something similar is sort of happening in hardcore optics. No, that I mean, so people we've been developing methods for optical imaging of neural circuits and manipulation of neural circuits and whatever. We do holography, and someone else does a light sheet and something else. And it's becoming clear that the future is hybrid. It's like a hybrid future, no? Because you'll use several of these tools together. And this has not yet happened. I think in a way we're sort of conservative. We all stick to what we know. But maybe this is we should try to do something to shuffle the the methods. Maybe send people from one lab to another lab. To infect people, to sort of cross-infect each other, no, with local expertise. I don't know. It's just an idea.
One thing that I've been noticing in the very recent past, I went to the Gordon conference in Plasmonics a couple of weeks ago in Maine, and I found 4 groups working in neuroscience. And that, I mean, I come from the field of plasmonics, nanophotonics. And I was very surprised because I'm also working with Rafa in neuroscience these days, right? But one thing that I noticed is that many people, to some extent, they try to do neuroscience, but they stay very much on their niche. And so basically they develop what they know very, very well, but then they put some neurons on top. You know what I mean? And I couldn't find many people who really established a network with serious, let's say, well-established neuroscientists to do their experiments on slices, to do their experiments in vivo, to do their experiments in real neuroscience problems. Like what I felt is that we were basically what I saw is that people were trying to measure synaptic currents or induce firing of neurons, but very simple things that to us are super exciting because we know nothing about neuroscience. But I missed that connection of being brave enough, but of course it requires a lot of compromise, as Rafa and I know from experience. Now we've been collaborating for 4 years, and it's a lot of work, training people from a different field, etc., etc. But I don't know. I do think that there is plenty of room for exploration. I don't know if for discovery, but at least for exploration. Right?
So in fact, this here's a question for Shimon, who's been actually in this field between NanoNeuro maybe longer than anyone here. No, given all this I mean, fantastic opportunities. Why hasn't it yet happened? How come we're still in 2022 and I mean, you have quantum dots, nanoparticles, nanoprobes, and they're just a trickle of neuroscience work. Why hasn't it happened? Yeah.
I think it's a bigger question. I think quantum dots have not really delivered on the promise over 25 years, really.
But in biology or in physics?
In biology. In biology.
Yeah.
In biology. You know, you can buy TVs, right? But in terms of making a huge impact on biology, you know, initially the promise was it's a fantastic replacement for dyes.
Yeah.
That did not happen for many, many reasons. I mean, it's slowly trickling and there are some good applications, but it's not replacing dyes, right? And I guess the same for specific applications in neurobiology. There will be some niche applications, but it's not one tool fits all. And there are many issues: stoichiometry, surfaces, blinking, size, many, many reasons why it's not a replacement. But there are some issues.
Has anyone ever tried to get quantum dots into the brain using the ultrasound approaches?
I wouldn't know.
They might be a little bit big, so yeah.
And as a result, you know, people really switch to polymeric, biodegradable quantum dots, right? Equivalents. My discussion on nanodisk, this for me is just an organic quantum dot in a way. Very simple to engineer and build, and the inorganic quantum dots, I think, will have applications in biology, but it's not a replacement. It's not going to take over. But you asked about combinations, specifically for things I discussed, for example, protein engineering to anchor inorganics into membrane.
Yeah, I mean, also Dirk was genetically engineer chemical probes. No, I think this seems to me like a win-win situation, because it's very clear that I would say biology today, the last 50 years, has been molecularly driven. So there's just an enormous amount of rich developments. You just heard we just heard Alice as an example of this, no, and tapping into that by linking nanomaterials or chemistry.
Pharmacology, right? The dream of administering drug at the place and at the time that you want.
Yeah.
So.
Yeah, that is. I mean, Jerry, I loved your to put things in place by showing us how little money is spent on research compared to Netflix. No? But as a former medic, I think it's really embarrassing that in 2022 we treat patients by giving them a drug that wipes out the body, that goes through the entire body, and even if you have a problem with the little tip of your finger, you still have that high concentration of agent anywhere in the body. I mean, we should be able to do better than that. I mean, we have methods to. I mean, besides sending people to the moon, and we do all kinds of fascinating things with light. Not so it would be an obvious thing to have like an optical medicine in a way, no, as Dirk is saying, an optical pharmacology.
It doesn't have to be optical. It needs to be, as Alice said, it has to be Boolean logics, where you get to the right place, and if component A and B are there, you release the content of a nano vehicle or something.
Yeah,
could be light. It could be.
Speaking of gating, new modalities. What about X-ray gating? Are there proteins that are super sensitive towards radiation damage or reactive oxygen species? I mean, at X-ray at the intensities that are used for imaging, what can you do with this? And it's very hard to break bonds that way in a selective fashion. Although there's some stuff starts to emerge in the literature. But
there is some stuff. You can do scintillation, right? You can actually use things like quantum dots and similar nanoparticles and go all the way from hard X-rays to visible, and then do something in the visible.
But there's not a catalog of hypersensitive proteins that are extremely sensitive towards radiation damage, with whatever mechanism I cannot think of. But has anybody cataloged this? I mean, from X-ray crystallography, this should be kind of known, right? The intensities are way out of.
But why should it be so selective?
That's the question, right? So is there any, I don't know, arrangement of side chains that makes a protein particularly sensitive towards radiation damage, or reactive oxygen species? I mean, it's probably the more likely case that you generate hydroxyl radicals or whatnot, and then you engineer a protein to be extremely sensitive towards that with some functional readout.
Mm-hmm. Anyway, we're just treating people now exactly the same way that we treated people 100 years ago in terms of pharmacology. I mean, it's just ridiculous, no? I mean,
I don't entirely agree.
Oral.
You know, Abraxane was the first nano drug that was delivering, you know, Taxol in a more specific way than just through the body, right? It was to some extent targeted.
Mm-hmm. Yeah, no, but I mean, how about 99.99% of all patients today in the world?
No, sure,
sure. So,
but
there's something wrong with this picture. It's been 100 years of science.
But many people are trying to build delivery vehicles that are targeted and specific, and you release the drug when you want, where you want, right? That's the vision. We are not there yet, but people are working on this.
Mm-hmm.
At least we're not doing lobotomy anymore, so there's some progress.
Well, let me tell you, for intractable epilepsy, they take out that piece of brain. So, I mean, this is in every big hospital in the world today. I mean, over and over again. I mean, you have patients that 30% of epileptics have they don't respond to drugs, partly because how in a primitive pharmacology is the way. So the only cure for these people is the scalpel. No? It's just terrible.
Yeah, there have been a lot of advances, you know, in more sophisticated, you know, therapeutic approaches in the cancer space. It's just, you know, with engineered cell-based therapies and, you know, protein-based therapies that are, you know, more clever.
But Alice, your end gates can be implemented not only for recording, they can be implemented for releasing drug at a particular site, right? That Boolean logic idea could be used to activate a release.
Right. I mean, I think you know, the main challenge with those types of genetic tools and actually trying to use them in any kind of translational context is immunogenicity. Right? Is that we're, you know, using components that are not all derived from the human genome, and they'll be recognized as foreign. We're using components of viruses, and so I think that's a deal breaker, you know, if we're trying to use that in any type of translational setting. You know, there's a lot of efforts of people trying to take these, you know, engineered synthetic circuit type approaches and overcome those types of things. So, for example, SynNotch is a technology that came from Wendell Lim's lab and has been used in CAR T cell therapy. And Kyle Roble at UCSF is trying to make them more humanized and, you know, external control so you can, you know, switch them off when you need to, you know, with the hope that they can actually be implemented in therapy. So yeah, you know, our tools are not designed with translation in mind at the outset, and so we take components from, you know, all types of species.
So here's a question for Jerry. How about using this ultrasound opening of blood-brain barrier with your viruses therapeutically? So have you guys started exploring this, or is anyone doing some clinical trials?
A little bit, actually, yeah. Yep. So there's a technology I didn't mention because we're out of time. We actually have a clinical trial that we got funded recently on using focused ultrasound without the gene delivery, but rather to recover proteins from the brain into the blood So you can have like a non-invasive brain biopsy. So the ultrasound has been used in a number of patients. I want to say about a couple hundred. So a few people are trying to use it. For the viruses, there's a lot that has been done recently with AAV gene therapy clinically, and a few of them were FDA approved. So technically, you have ultrasound that has been done in humans, and you have the viruses that have been done in humans. So now it's just a matter of doing them together. So I think that there is a good chance, right? We're also having some projects that focus on bridging the gap to test whether it's worth it, right? So if you spend $500,000 on a virus, deliver that to the brain, then find out that actually doesn't work, then that's gonna suck. So we're working on some tools to prove it or validate it without having to gene therapy to see whether the neuromodulation, chemical neuromodulation can be effective. So I think it's gonna happen. It's probably gonna take a moment. We're also working on making those viruses, actually, the viral vectors for brain delivery, to make them work in humans. So we have we're negotiating a collaboration with a pharma company that I don't think I can name yet. We're gonna try to make viruses that you've seen, but for humans rather than mice. Yeah, so it's inching forward. Eventually, I hope it's gonna happen, but probably not very soon.
So actually, if you have a stroke, a local stroke, the blood-brain barrier breaks up locally. So this could be also, stroke could be a disease in which many of the tools that you and all of you are developing could be first tested as a translational avenue, into the brain, because you don't have to worry about the blood-brain barrier, and then there's a way to rescue these neurons that are damaged after stroke. I mean, what do you think of that, Jerry? Have you thought of stroke specifically, or anyone in the panel or in the audience?
A little bit, but I think that there's a lot of potential end case that could work for stroke because it's so complicated. So I think there's other tools that would be better than those AVs.
Okay, there's a question from the audience, but it's pretty much what we've been talking about. It's are any of the tools discussed moving towards application in humans for human neuroscience research or therapeutics? If not, could you discuss challenges for moving towards human neuro applications? I guess this is just to highlight this issue that somehow we're not moving the needle much in the patients, and at the end of the day we're all biomedical researchers.
But Rashi, you said that focused BBB opening has been used in humans. Did I understand this correctly? This has been applied to humans.
Yes. I might have not been clear, but let me show the data.
This is considered safe.
That's been done a number of times in different diseases, but this is actually a human head that you're seeing here with BB opening. Yeah. So people found that just opening itself can have some therapeutic effects in mice. So sometimes they don't even deliver anything. But it's been done for brain cancers. It's been done for something like PD, I think, Alzheimer's disease. There's a few trials that are ongoing. Yeah.
And what is the mechanism of the opening?
So people have different ideas about it, but the leading hypothesis is that the contrast agent that is compressible with ultrasound gets compressed and then expands when the ultrasound rarefies. So it just begins to oscillate with ultrasound and physically presses on the blood vessels to loosen tight junctions. So you dial parameters so it doesn't burst the cells, but just loosens tight junctions between them. And you can get a particle up to about 25 nanometers. The biggest I've seen is about 50 nanometers, but the efficiency was low, right?
Mm-hmm.
So that's how. And you can focus ultrasound like light, right? So just like you focus the light, you can actually focus ultrasound waves down to diffraction.
And how dangerous and also how expensive is this approach? Like, is there a danger of, power is too high and,
Yeah, you can overdo it. You certainly can overdo it. So there's like this threshold actually of cavitation. If you go beyond that threshold, the bubbles, instead of oscillating nicely, oscillate bigger every time and eventually burst. And that burst is very, very dangerous. So it can actually break the blood vessels and cause localized hemorrhage. Now, the localized hemorrhage usually is just like red blood cells embedded in parenchyma rather than just blood flowing. But it's, it can result in neuronal loss and so on. So there are methods to monitor that you are not quite at that level. So there's this acoustic monitoring that actually can get an echo and see whether you're overdoing that. So that's what Kullerbach and Lisa Konopag here at Columbia and lots of other people spent a lot of time working to make those parameters safe. And it seems that they've gotten that to the point that they can do it. There's a device that kind of does it for you from Insightec that measures this cavitation echo and can open the brain using 1,024 different ultrasound elements. And as a result, it's very expensive. So it's like a couple $1 million. But medical centers have it. So Houston has one, Utah has another one, Swedish Medical Center has another one. So it's expensive, but it's workable. And some people, Santonius was in Elisa Konopag's lab, actually made one that is much cheaper, that allows you to just have a single element for a fraction of the cost. So it's pricey. I think it's gonna get better.
I wonder if the Havana syndrome is focus ultrasound. Do you know that, Jerry?
It's possible. Yeah, it's possible.
Yeah.
I read a little bit about it, and it seems like there's three hypotheses: that it's electromagnetic waves, that it's ultrasound, or that it's blast. So who knows? Yeah, who knows?
I would appreciate hearing from Rafa and other practicing neuroscientists, like, you know, what are the most game-changing tools that you would like to see developed?
Yeah, I think, I mean, I have a particular point of view, and that's why I've worked with 2-photon in my career. And that has to do that the brain is not just like the liver, but it's an organ built with probably at least a cortex with hundreds of different cell types, and they're connected in particular ways. And each neuron is different, and each part of the neuron may be different. So we really have to go down to the precision of the system in order to understand it and manipulate it. So we need tools that have single cell precision or even subcellular precision, ideally in the same time frames that nature is actually operating. So I think, I mean, I love what all of you are doing, and the previous panel. I mean, for me, this is like a feast, like going to a good restaurant of exquisite food. No? And I mean, I would encourage every one of you to continue in the same direction, but keep in mind, if you can tie this up with 2-photon or with holography, so that imagine like playing the piano with the neural circuit or different spines by inactivating spines or activating spines or turning on this protein here or there, and of course to do that in vivo and with the goal of moving to the human at some point. I think that's sort of my dream, not to simply to be able to read and write information in the brain with single synapse precision in vivo. I think that would be a dream. Yeah. I mean, you also measuring voltage. I have to tell you too, as one of the people that started using calcium imaging to monitor neuronal activity. And by the way, I have to tell you that we found this by chance. And Roger was in my thesis committee when that happened, and he was like looking over our shoulder all the time, like, what are you guys doing next? And we were at Rockefeller. He used to drop by the lab every time he came to New York to see what was up with this business of calcium and neural activity. But I mean, it's been very useful, and it's, I would say, been a game changer for neuroscience to do calcium imaging, but it's not the answer to all of our prayers. It's slow, it has all kinds of problems, and what we need to do is measure voltage, and that's still, I mean, we didn't have, we had Shimon talk today, and earlier there were talks on voltage, and there are a lot of JEVIs that have been developed, but we're still not out of the woods there. I think we still need at least an order of magnitude better measurements of voltage in both in amplitude and in temporal resolution, to make voltage imaging the bread and butter of neuroscience. I think which is what it should be. So yeah.
And we would love to design voltage integrators rather than calcium.
Yeah, exactly.
Just
exactly.
get, well, much
so
more directly.
there you go. Imagine if you did a voltage integrator and you could also see inhibition, not just excitation. I mean, that would be
absolutely GABA integrators. That's a holy grail.
Yeah.
Yeah.
Yeah. This is going to keep us busy for a while. These challenges, even with the brain initiative going on, this is bigger than the brain initiative. These issues.
We have a comment from an audience member saying that it would be really helpful to have robust methods to label and manipulate activated synapses rather than cells.
That's right, exactly. I mean, this is sort of what I was referring to. If you could do single spines, the spines to me is the same thing as a synapse. So if you can turn these methods, have them with a single spine precision, you can do that with 2 photons. So if your methods work with 2 photons, you're in. You can do that. No?
So I mean, so the channel rhodopsins plus 2 photon, that's not sufficient?
Channel rhodopsin, it's okay, and we've used it. We've actually collaborated with Carl with C1B1, and in that paper in 2012, we showed that you could actually turn on single spines with optogenetics, and we've used this recently as well. Okay, but I mean, for instance, I mean, how about talking to Dirk? I mean, if I can turn off CAM kinase 2 in one spine that I know has been activated with calcium voltage imaging in a particular type of behavior, I mean, something like that would be just a dream we're still scratching the surface in terms of the bringing these tools down to the synaptic level.
Dirk, are your PHOTAX azobenzene switches controllable by 2 photon approaches?
We haven't looked into this yet. I'm not equipped to investigate this. I only referred to previous work with 2 photon activation of azobenzenes. And azobenzenes are pretty good. They have a pretty nice 2 photon cross section. So we've done with David Di Gregorio, for instance, we made a photo-switchable glutamate to activate NMDA receptors, and we could beat diffusion that way by activating it and immediately inactivating it locally. This was faster than diffusion. So it was a Nature Communications paper some 10 years ago with Davide.
I see. But I guess one question is, if you were to do single spine, manipulation of CAMK kinase using your approach, what would be the temporal resolution? Because it takes time for the degradation to occur, and then you can have new molecules coming in.
Yes, it takes time, but it doesn't take that much time. So we see effects after, I would say, 12, 15 minutes. We see effects already. So it's not ultra fast. That's true. It's not at the level of an ion channel. That's for sure. But it's not. It doesn't take like 6 hours for it to. And where it happens, where the proteasome is that does this, that in itself is an interesting question to me. Right? Where this physically happens, the degradation. If you, I don't even know whether the proteasomes are in a synaptic spine. Right? So is this known? Whether they must be there, right? They must be pretty close. But where exactly are they positioned in neurons? And I'm not talking of the transmembrane proteasomes. I'm talking of the regular dumpsters.
Okay, so I think we're pretty much at the end of the meeting. So if this is okay with you, Sol and I can now.
I want to thank Rafa and Sol very much. That was a terrific day. Thank you. And thank you for inviting me.
Thank you. Well, thanks for your wonderful, stimulating talks. And yeah, I think our plan is to continue to do these meetings every year. We welcome ideas about topics that we may not have covered, or topics that are emerging that you think could be particularly interesting, not just from you speakers, but also from anyone in the audience. They know how to find us. They can just send us ideas. We're going to start working on next year's program soon. And yeah, and just to highlight that that this was also possible thanks to the Chen Foundation, which is based in Caltech, and they gave us funding to make this possible, and also the work of people under the hood, including Lux, James and Shilpita, and Javier and Teresa. And Anne, I think they should all be recognized that have contributed to the helping assembling the program, preparing the schedules, sending the invitations, and also manning the IT, and particularly with Jose. Am I leaving anyone else out, Aitzol?
I was going to say Jose Caballero, who rescued us today wonderfully. So he's going to watch this. So, thank you. And thank you all. Thank you all the speakers for saying yes to our invitation and for delivering such amazing talks. Once again, it's been a pleasure and super exciting afternoon.
So let's close this in Basque, since we're in the Basque country, Aitzol and I, and yes. You would say Bukatu al dugu, is that how you would say it?
Oso ondo, Rafa, bukatu al dugu, bukatu dezakegu.
Bukatu al dezakegu. So with that, those words means let's close it. Let's close this.
Thank you.
Thank you, and have a great evening or day. Take care.
See you soon.